and we would go forward. I'm new to the chair, so I guess I'm supposed to read this little spiel, and I'll go through it. Good morning, ladies and gentlemen. Welcome to the Anchorage for the Hatchery Committee meeting of the Alaska Board of Fisheries. I'm John Wood, chairman of the board and this Hatchery Committee. Today is October the 14th, and it is 8:30, whatever I said it was, 4:06. Start out with general information, including hearing disabilities. Yes, I was going to ask you to do that.
Yeah, just if there's anyone in the audience that has a hearing disability or just needs a little extra help, hearing what's going on, we have some assistive headsets that tie into our sound system. So if anybody wants to borrow one of those, just see our staff at the end of the table, and we'll get you set up with that.
Okay. And at this time, I'd like my fellow board members and staff to introduce. We'll start with the board. From my right-hand side, Mr. Wood.
Mike Wood. Full introduction? In case you don't know me, I'm new at this. I'm from north of Talkeetna and Chase, and new on the board here. And are we supposed to read our disclosure thing?
Negative,
just
not for this meeting. This is not a regulatory meeting.
All right, I'll end it at that. You can grab me, and we can talk anytime you want.
Okay.
Thanks.
Mr. Swenson.
Greg Swenson. I'm also new on the board and was born and raised in Anchorage. And same thing, if you have things that you'd like to talk to me about, I'm very open to hearing what you have to say. Thank you.
Good morning, everyone. Tom Carpenter from Cordova.
And John Wood from Willow, and I'm new to the chair, so we'll stumble through it together.
I'm Stan Zaray from the village of Tanna on the Yukon River. Thank you.
Jared Godfrey, Eagle River, Alaska.
Good morning, everyone. Mark Carlson Van Doren. I live in Anchorage, grew up in Juneau and Chesapeake Bay. Thank you.
Thank you. Commissioner?
Yeah, good morning. Douglas Lang, Commissioner, Alaska Department of Fish and Game, and I will let my staff introduce themselves and their staff.
Good morning, everyone. Israel Payton, Director, Division of Sport Fish, and with me here today is our Deputy Director, Tom Talby, behind me. We have Matt Miller up against the back wall. He's the Cook Inlet Regional Management Coordinator. We also have Patrick Fowler from Southeast. He's the Regional Management Coordinator. And today, giving the Sport Fish Enhancement presentation will be Chuck Pratt. And he's up against the wall there at the table. Thank you, Mr. Chair.
Thank you. And subsistence is not here today, correct? No. Okay.
Yes, that's correct. I did not think that this was something that we needed to hold somebody here back for our subsistence.
And Department of Law?
Good morning. I'm Noah Starr with Department of Law today.
And Wildlife Trooper, Captain DeGraff will be here or not? Does anyone know? Commissioner?
I don't think he will be here, but we have Sam Raybung from Commercial Fisheries that needs to introduce his staff.
So we'll pin a badge on Sam and run with that. Okay, sounds like a plan. With that, we'll proceed. And we have a joint protocol on how this meeting is to be conducted and what's to be done there. It'll be first.
Sam didn't introduce his staff yet.
Oh, Sam, how could I forget you? It's a Hatchery Committee meeting, too.
Way down there at the end of the table.
Sam, please introduce your staff.
Good morning, Mr. Chair, and members of the board. For the record, I'm Sam Raybung. I'm the Director of the Division of Commercial Fisheries. And with us today is Forrest Bowers, our Deputy Director, Mr. Bill Templin, our Chief Fisheries Scientist, Dr. Katie Howard, our Salmon Ocean Ecology Program Lead Scientist, Sarah Gilk-Balmer, our Gene Conservation Lab Director and Principal Geneticist. And online with us, because weather prevented them getting here from Juneau, is Lorna Wilson, our Assistant Private Nonprofit Hatchery Program Coordinator, and Mr. Flip Pryor, the Aquaculture Section Chief for the department. Thank you.
Thank you. By way of introduction, Mr. Pappas.
Good morning, Mr. Chair. George Pappas, Office of Subsistence Management, Housing, Fish and Wildlife Service. It's great to be here. Thank you, Mr. Chair.
Okay, thank you. Art, you want to?
Good morning. My name is Art Nelson. I'm Executive Director for the Board of Fisheries. And with the board staff we have here today, at least for just a little bit longer, Annie Bartholomew, Savannah Hollingworth, and Farie Fernandez. Mr. Chair.
Thank you. As mentioned in the joint protocol, there will be no regulatory action and no hatchery-related petitions of agenda or agenda change requests will be considered as action items during today's meeting. The meeting today is a committee meeting, not regulatory. The board always encourages, appreciates additional written comments and receive them from the public today. But our process of receiving them will be unlike that which occurs at a normal regulatory board meeting. Anyone attending this meeting who would like to submit written comments to the board for its consideration and can submit them through the board's website, or you can turn them in here at the meeting at the end of the table to my right. Please note that you only need to turn in one copy, but board staff will not be making copies in color. So if you have something in color, you're going to have to do it on your own, and you'll need to return in 20 copies of those color articles. Individuals may also submit written comments by fax to 907-465-6094. Written comments will be assigned an RC number and distributed to board members, and posting to the website will occur periodically as staff time permits. We'll now go into staff reports, but before we do that, the director and I try to figure out which order you're going to present them in, and I'm going to have him read it off as to what we believe to be the order, but if that's incorrect, we need to change it accordingly. So we'd start off, well, I'll do it. We'll start off with a report to be given by Dr. Howard, which is the overview report, followed by the next report to be given by Lorna Wilson, which is the Fishery Enhancement Program planning efforts, then followed by the Sports Fish by Chuck Pratt, then followed by the Sarah Gilk-Balmer report on the Hatchery Research Program findings, and then the last two reports by Bill Templin. I don't know which order he's going to present them in, both of them related to Hatchery Research Program. So if that's not the order, I need to know that going into it. Otherwise, that's how we will proceed. Okay, so whoever's,
Mr. Chair, on the last three, I think we're going to do a little different order.
okay.
Which order is
This one,
Okay. The Alaska Hatchery Research Program overview, and then Sarah Gilk-Balmer's presentation about findings, and then followed at the final is the making decisions presentation.
Thank you.
Thank you.
So noted. So if Dr. Howard would come forward, I saw her hand up, so I know she's here. Yeah. Why don't you do that since you're more familiar?
Sure. Board members, one of the first things that we typically do at the beginning of the Hatchery Committee meetings is just a quick review of the joint protocol between the board and the department on salmon enhancement. This is a finding that the board adopted in 2002. It's RC1. It's also posted to the meeting page, and it just kind of lays out the intent. And if you're fine with this, Mr. Chair, I'll just read it. It's relatively short. In actions taken in January 2001 and June 2002, the Alaska Board of Fisheries stated its intent to institutionalize a public forum to bring statewide perspective to issues associated with hatchery production of salmon. Accordingly, the department and board agreed to enter into this joint protocol to coordinate department and board interaction on certain aspects of salmon hatchery policy and regulation. Authorities. The Commissioner of the Department of Fish and Game has exclusive authority to issue permits for the construction and operation of salmon hatcheries. The Board of Fisheries has clear authority to regulate access to returning hatchery salmon and to amend by regulation the terms of the hatchery permits relating to the source and number of salmon eggs. The Board of Fisheries authorities also include the harvest of fish by hatchery operators and the specific locations designated by the Department for harvest. See Alaska Statute 1610, 440B, and Departmental Law Memorandum to the Board dated November 6, 1997. It is the intention of the Commissioner of the Department of Fish and Game and the Chairman of the Board of Fisheries that meetings be held on a regular basis wherein the Department will update the Board and the public on management, production, and research relating to Alaska's Salmon Enhancement Program. The Joint Department Board meeting on hatchery described here will take place at a mutually agreeable time and place during regularly scheduled meetings of the Board. The meetings will provide a forum for open discussion on a mutually agreed upon agenda of hatchery topics. The agenda may include site-specific as well as regional or statewide hatchery issues. These salmon enhancement meetings will not be open for regulatory actions, and no hatchery-related petitions or agenda change requests will be considered as action items. These meetings are open to the public at its discretion, and upon appropriate notice, the board may open the meeting to public comment. The hatchery meetings will provide an opportunity for the board and the public to receive reports from the department on hatchery issues, including production trends, management issues, updates on hatchery planning efforts, wild and hatchery stock interactions, biological considerations, and research. Requests for report from the department may be made during the board's work session during meeting years when there is a hatchery forum scheduled. As appropriate, the board and department may agree to invite other state and federal agencies, professional societies, scientists, or industry spokespersons to attend and to contribute information on particular topics or sponsor other discussions such as marketing or intrastate effects.
Thank you.
Mr. Chair.
Thank you. Any board member have any questions as to understanding of what the protocol calls for? Seeing none, we'll proceed. Staff reports. Dr. Howard, you're first up. You have the floor.
Good morning. For the record, my name's Katie Howard. I lead the Salmon Ocean Ecology Program at Alaska Department of Fish and Game. And this presentation will provide a brief overview of our current scientific understanding of salmon competition at sea. This is simply a recap for those of you who saw my presentation at the last Hatchery Committee meeting. The remainder of the presentation is updating the board on research the department is undertaking that either directly or indirectly is helping to fill some of the information gaps for the topic of salmon competition at sea. The Salmon Ocean Ecology Program is intended to provide information on the marine life of salmon that can help inform management decisions. Part of my job is to gather information across the scientific literature and from our own research, and to summarize it in as unbiased and open-minded way as possible and share that with decision makers like yourselves. Sometimes there is clear scientific advice I can provide based on these summaries, and sometimes it is less clear. Salmon competition at sea is a topic that falls under the latter category. If you ask 3 different scientists to come talk to you about this topic, you might get 3 very different perspectives. And this is because this topic is one of intense debate among the scientific community. There are many opinions about salmon competition at sea from individual scientists, but not an agreed upon opinion of the scientific community at large. Just like I'm sure probably exists within this room, there are some very diverse perspectives in the scientific literature. Some bold statements are being made on either side of the issue, and a lot of gray area in between. I pulled out just a few quotes to exemplarize how polarized the debate is. On one side, we see statements that pink salmon in particular are responsible for a resource vacuum that has a destabilizing effect on the ecosystem. But on the other side, you see statements that these kinds of statements are seen as fantasies or absurd. And the one quote I think many can agree on is the one in the middle, that the potential for food resources to limit salmon production across the North Pacific continues to be vigorously debated. Now, in general, the scientific literature on salmon competition between different species at sea falls in 4 categories. And just to be clear, I am only speaking to scientific literature in English written journals. There are many Asian scientists who publish in non-English written journals, and most of us North American scientists don't have access to those in part because of the language barriers. But in English written journals, we see suites of papers that are focused on diet overlaps and diet shifts, utilizing a variety of methods to investigate which species and stocks have the most similar or dissimilar diets and the highest or lowest likelihood of potential for competition. We see suites of papers that in one form or another look at relationships in the abundance, productivity, or survival trends in species that might compete with one another, investigating whether one species is in a poor phase at the same time as another species is in a strong phase. We also see suites of papers that are focused on growth with the assumption that growth is related to marine survival. In these, higher abundance of a competitor species is associated with lower growth of another species. And the last suite of papers we often see are those that associate competitor abundance patterns with age at maturity, where high abundance of a competitor species is associated with earlier maturity of another species. Now, typically, the evidence that salmon diets overlap, which is shown in category 1 papers, is suggested as the evidence that competition is possible, and the studies in categories 2 through 4 is presented as the evidence that competition between salmon species with serious negative consequences is occurring. The nature of the methods, particularly for categories 2 through 4, is where a lot of the debate comes into play. The primary arguments made on both sides of the issue are scientifically valid. For those who are convinced that observed patterns in Pacific salmon abundance, survival, and size are clearly due to competition at sea between salmon species, they argue that direct assessment or experimental studies in large marine ecosystems are incredibly difficult, and so indirect evidence is necessary and can be considered sufficient because very similar patterns are consistently found across multiple situations. These perspectives largely focus only on salmon as the potential competitors. In these perspectives, there is often a focus on pink salmon with the view that the biannual life cycle of pink salmon provides a natural experiment that can be leveraged for analysis. This perspective largely draws from English written journals and North American research approaches. For those who are not convinced that this suite of research successfully demonstrates that observed patterns in Pacific salmon is caused by competition at sea between salmon species, they point to a reliance on indirect evidence as being a weakness. They point out that studies that encounter a lack of relationship are often ignored by researchers and are not published. And in science, we call this confirmation bias, and it exists across all research. They consider the focus only upon competition between salmon species as short-sighted and ignores the roles of other non-salmon competitors in the marine ecosystem. And they point out that while salmon are very important to humans, their importance as predators in the marine ecosystem is relatively small when considering other species with similar diets such as pollock, cod, and herring. They argue that not enough has been done to rule out alternative explanations for the correlations that are observed. And for example, those biennial patterns are known to occur for some other species as well, like squid and some copepod species. And as I alluded to earlier, there is a disconnect between the kinds of data and perspectives of North American scientists and those of Asian scientists. Both Japan and Russia have a long scientific tradition of conducting these really large marine surveys to study salmon. In fact, the marine surveys that we have in North America now are largely modeled after what Russia scientists have been doing for many decades. As you can imagine, Asian scientists more frequently use that survey data to inform their analyses and directly investigate issues of salmon competition at sea. And they tend to put it more in the perspective of the whole ecosystem, while North American scientists often don't have those data sets, and so they're more frequently relying on models to indirectly investigate salmon competition simply because those data aren't available. The reliance on these very different tools leads scientists to different perspectives and conclusions. And so that does beg the question of how do we move forward when there are these really polarized perspectives? One thing we can do is address the arguments head-on by filling data gaps and evaluating some of the assumptions. A major point of contention in the debate is about how much we should rely on indirect evidence that is primarily driven by correlations. There is some good guidance, as you see here, on when we can consider correlations strong evidence for causing the patterns that we see. And for the most part, the literature on salmon competition at sea relies pretty heavily on the second point here. There is a consistency of pattern across multiple situations. But there is more that we can do to address these other criteria and provide more clarity for how heavily we should weigh these correlations. We can investigate competing explanations, for which several have been proposed but not adequately studied. This would either rule them out or elevate them as more probable explanations. We can also gather more direct observations for North American salmon, which would allow us to address point number 4 here. And so what I'm going to provide here is just a hypothetical example of the kind of work that could be done to provide some of this clarity for this topic. So right now, we may have an observation that when species A increases in abundance, there has been an associated pattern for species B to decrease in abundance. And perhaps the conclusions that the authors drew from this observation is that hatchery releases of species A has contributed to the decline in species B. Now, if we use that guidance in the previous slide, for when we might consider correlation strong evidence for causing the patterns we see, we might want to look for what we call mechanistic studies to support this conclusion as an appropriate interpretation of these observations. And so mechanistic studies is just scientist lingo for investigations that demonstrate that the components or the mechanisms in the system are there that would need to be true in order for the correlation to be indicative of a causative effect. So to give you some examples, in this hypothetical instance, one mechanistic study might be to look for evidence that species A and species B are in fact relying on the same food source. So they're using the same habitats at the same time and eating the same things in those habitats. That provides some additional support for this conclusion. We can also look for evidence that the prey base is insufficient to support the biomass of both species. So this is one type of mechanistic study that has been done in the Western North Pacific by some Russian scientists, where there is this long history of marine surveys assessing salmon and their prey. But that really has not been well studied in the Eastern North Pacific. We can also see if we can rule out other potential causes for the correlation, such as both species A and species B are interacting with a third species that is causing them to have these patterns, or that they're reacting differently to shared environmental conditions. We can also investigate whether there is some characteristic of species A that gives it a competitive dominance for food over species B. We would also want to demonstrate that the increased biomass of species A is because of the hatchery releases rather than wild abundance fluctuations, in order to clarify that link to hatchery fish specifically. And additionally, an observation that species B is starving and species A is not would be another piece of evidence that would support that this conclusion is a correct interpretation of this observation, and there is a causal link there. As you can see, multiple pieces of these mechanistic evidence, these studies, would provide more and more strength to this conclusion. Unfortunately, this kind of mechanistic evidence, those things in these colored circles here, that's what we're generally lacking now, especially in the Eastern North Pacific. But it is the kind of information that we can work towards contributing to move the science forward. Now, over the next few slides, I will talk about some of the efforts we're involved in to attempt to fill some of those data gaps and provide more clarity to this issue. Some of these are studies trying to get direct mechanistic observations, like referenced in that previous slide, and some are focused on enhancing the contextual information that we have and is used in many of the existing correlation analyses. Current efforts I'm going to talk about are leaning heavily onto international collaborations, as you will see. The North Pacific Anadromous Fish Commission, or NPFC, is an intergovernmental organization established by the Convention for the Conservation of Anadromous Stocks in the North Pacific Ocean. ADF&G staff are among the U.S. delegation to the scientific working groups that contribute to the NPFC's mission. One of the new products produced by the working group on stock assessment is a Pacific-wide synthesis of stock assessment information across the Pacific. This is the first report of its kind, and it will be published in this fall. This figure is one example from that upcoming report, showing changes in pink salmon commercial harvest in recent years relative to historical years. The colors represent the degree to which catches have increased or decreased relative to historic levels. And this Pacific-wide synthesis provides a better understanding of which regions are seeing increasing or decreasing abundance. Although this product is not assessing competition at sea per se, this and other efforts of the working group are building and improving our understanding of Pacific salmon abundance patterns and trends across their entire geographic scope. And this is important contextual information that is used in assessments of competition at sea. For example, we know that not all salmon stocks use the same habitats when they're in the ocean. So regionally explicit index information helps us understand which stocks are driving the overall salmon abundance patterns. A similar effort we are working towards within that working group that will help inform this information about the role of salmon competition at sea is to improve abundance estimates for salmon at sea Pacific-wide. There has been some pioneering work by Greg Grugeroni and Jim Irvine to create Pacific-wide estimates of salmon abundance at sea. And this figure you're seeing here is from one of their papers, illustrating the adult and immature salmon biomass of sockeye salmon in red, chum salmon in yellow, and pink salmon in pink since 1925. Many, if not most, of the North American salmon competition studies use these estimates of salmon abundance at sea in their analysis. However, this effort that's being put forth by the NPAFC working group is attempting to get member countries of the NPAFC engaged in taking on the responsibility for building upon and improving these estimates with additional data, and by making those estimates available to researchers on an annual basis, which isn't the case now. So this should help enhance salmon competition analyses overall. There are a couple of notable efforts related to salmon competition at sea under the International Year of the Salmon, or IYS, initiative. The International Year of the Salmon effort was a 5-year initiative with a core focus on collectively generating and sharing knowledge across the international community in support of sustainable salmon. The IYS initiative included a variety of components. One of these was to convene a group of experts across the Northern Hemisphere on pink salmon. ADF&G staff participated in this diverse and important dialogue about pink salmon, and that discussion ranged broadly across the Northern Hemisphere, so both within the Pacific and among our colleagues in the Atlantic who are grappling with pink salmon as an introduced species. That discussion included an improved understanding of how pink salmon abundance is changing, approaches to assessing and monitoring pink salmon, competition and interactions between pink salmon and other species, genetic stock delineation of pink salmon, and approaches to monitoring and predicting future changes to the marine distribution of pink salmon, among other topics. The work of that expert group is reported in the published technical report, and I've added a link for that to this slide. This map is from that report, and it depicts where current spawning distribution is, and the current spawning distribution is shown in green. The green hatched areas on this map are locations where run abundance and catches dropped dramatically in recent years. The orange areas are depicting limited spawning distribution in major rivers. The orange hatched areas are those where regular occurrence of pink salmon have been observed in odd years since 2001, so that's more recent distributions. Red represents historical spawning distributions that is no longer used. The clear areas with orange hatching represents new areas of pink salmon occurrence since 2000, and blue represents, in the ocean, represents the total ocean distribution of pink salmon, including returning migration areas. And I would particularly point out that the component of that report that relates to competition at sea, you will notice, if you read that component, that much of the discussion in that paper is mirrored in the slides I've provided in the first part of this talk regarding the contrasting scientific perspectives on salmon competition at sea. Another component of the IYS initiative was to directly assess salmon during winter across geographically large survey areas. Again, this is in the eastern North Pacific, which historically has not had a lot of marine survey activity compared to the western North Pacific. This effort included surveys in the eastern Gulf of Alaska in 2019 and 2020, and these maps are showing survey tracts where salmon and their ecosystems were assessed in those years. The keystone project of IYS was a broad survey across the central and eastern North Pacific Ocean in winter of 2022, and this involved 5 ships and a tremendous degree of international collaboration. I have a link at the bottom of this slide that directs you to a recent publication that shares some of the data I'm reviewing with you today. Now, in collaboration with our NOAA colleagues and university researchers, we have received grant funding to leverage some of the data and samples collected from these surveys. Among other things, this project will allow us to investigate competition at sea in winter when competition is expected to be the highest. Because we went out there to collect samples, these will be direct observations. Our primary focus is to look at spatial overlap, dietary overlap, evidence of limited prey resources, such as physiological signatures of starvation, and other indicators of competition for food. And our focus is on particularly AYK chum salmon relative to other species and stocks, as well as non-salmon species. Otoliths from sampled salmon are also being processed so that we can identify hatchery fish within those potential competitors. Although laboratory analyses and data syntheses are ongoing, Lori Whitecamp has led a number of presentations and papers that summarize some key initial findings from the survey efforts throughout the IYS program. I will share a few of these data figures over the next few slides. One key finding from these surveys includes the relative catch and distribution of species identified in each of the 3 survey years. Chum salmon dominated catches in 2019 and 2020, while sockeye were very abundant in 2022 surveys. Now, many of these sockeye in the 2022 surveys were encountered near the Aleutians, and that was an area that was not sampled in those previous years. While Chinook and coho were expected to be relatively low in abundance, in part due to their overall lower abundance in the ocean, but also particularly for Chinook, they inhabit deeper habitats when they're in the ocean compared to the other salmon species. So the gear we were using to sample, we expected to find fewer Chinook just based on that alone. What was really surprising in these surveys that nobody quite expected is we didn't encounter as many pink salmon as we expected, because there should be a lot of pink salmon in the ocean in these years, and we just did not encounter them. They must have been overwintering outside of this region where they were expected to be. This figure shows the distribution of catches for each of the salmon species, as well as steelhead in 2022. The colors of the circles are simply representing which vessel they were caught on, but what's important is the size of the circles here, and that's showing the size of the catch in each of these areas. The bigger circles indicating larger catches. Particularly for chum and sockeye, the largest catches were observed in the central North Pacific, just south of the Aleutians. Genetic samples were taken from salmon collected on the IWAS surveys, and this figure is showing chum salmon genetic analyses. Chum salmon were sampled, were grouped from these collections from the central North Pacific, which if I can get, maybe I can't get the cursor to work. So that's the blue oval that you see on the map, and that is related to the results you see on the top left-hand figure. And we also collected samples from the eastern North Pacific, and that's the green oval on the map, and that's related to the bottom left-hand figure results. In the central North Pacific, the chum caught there in winter of 2022 were mostly Asian origin. So that includes Japan in light blue color and Russia in pink color. In the eastern North Pacific, the predominant chum salmon stocks originated in rivers that empty into the Gulf from the western Gulf of Alaska, southeast Alaska, and British Columbia. We can also look at the genetics of individual fish to better understand the distribution and migration patterns of particular stocks. So this is showing the presence and absence of different stocks sampled across the sampling region. This line is showing that division between the eastern North Pacific on the right-hand side of this map and the central North Pacific on the left-hand side of this map. Black dots show locations where we sampled fish, but no chum salmon were caught. The size of the pie graph represents the size of the chum salmon catch in that area. So these pies are scaled to the chum abundance at each station. I want to draw your attention to the dark blue color. So these are the Western Alaska chum salmon. That is the focus of this project, this collaborative project we have with NOAA and university researchers. Those Western Alaska chum salmon stocks are present fairly broadly from east to west across the southern end of the sampling area, with the highest concentrations appearing in the eastern part of the central North Pacific and the western part of the eastern North Pacific. So this provides us new insights into how these AYK chum salmon are migrating in the ocean, particularly in winter where we've previously had very limited information. It also shows us which other chum salmon stocks are in the same areas as AYK chum salmon and potentially competing with them for food. We can also look at other species that may be overlapping in space with AYK chum salmon. For example, this map shows pink salmon distributions. Genetically, pink salmon can only be broadly distinguished between Asian and Bering Sea stocks, which are shown in orange here. And then also Gulf of Alaska and Pacific Northwest stocks are shown in pink. Now, I just want to draw your attention, there was this one oddball fish that was identified that genetically is of odd year lineage instead of even year lineage. So this was potentially a fish, a 3-year-old pink salmon. But when we think about the overlap between pink salmon and chum salmon distributions, about half of the stations where the Western Alaska chum salmon were found also did observe pink, but the abundance of pink salmon were generally very low at these stations. Pink salmon are supposed to be the most numerically abundant salmon in the ocean, and our results do beg the question of where these pink salmon are overwintering because they weren't abundant in the sample region as what was expected. Ongoing work in these analyses will include looking at otoliths of the pink and chum salmon to identify hatchery origin fish, as well as work to investigate dietary overlap, physiological indicators of starvation and stress, investigations of the prey base available to salmon in this sample area, and a look at other potential competitors in addition to the salmon species. Now, what I've presented here are merely updates on ongoing efforts the department is engaged in, and we'll continue to work on these projects to address information gaps and add clarity to this issue over the coming years. We periodically provide updates on this and other marine salmon research through our reports, presentations, and on our Facebook page, which is accessible through this QR code. Thank you.
Thank you, and we're going to entertain any questions from board members. What I'm going to do, since there's so much material in front of us on this report, and I thank you for it, is whoever it is asking the question has a particular slide that they want to refer to. Once they get their questions satisfied, I'm going to stop and ask if any other board members have any questions on the same slide so that we can go in a more orderly fashion. And with that, I'll open it up to questions from the board. Are there any questions of Dr. Howard? Ms. Van Dort.
As always, Dr. Howard, thank you very much. Excellent presentation. My question is on slide 7, and when you were going through the observation and conclusion sort of continuum, and you made reference a couple of times to a lot more science that was being conducted by the Russians and Japanese, perhaps. What are some of the conclusions from some of those Western Pacific studies, that, if you're aware of any?
Through the chair, a lot of the work that they have done has been focused on using their marine surveys to look at the whole ecosystem. And so they're looking at, is the prey base adequate to serve all of the salmon needs? And their work is in the Western Pacific, so I just want to make that clear. And what are the food demands of the abundance of salmon in that region? And their conclusions so far, from what I've gathered, what I've been able to read, is that they haven't found evidence in the Western Pacific that the food base is limiting there.
Thank you.
And slide 7.
Katie, second to your last slide. And because I wasn't paying attention, I was curious what that highlighted yellow, next one, right there, what's that mean?
I'm sorry. Through the chair, so that one individual was genetically identified as an odd year lineage, which we assume was a 3-year-old fish. So it's a very unusual observation.
Thank you.
Yeah, and trying to act as traffic cop, I overlooked, I did have a question on slide 7, and it's really easy. Is there any significance to the circle's subject matter being above or below that black line?
Through the chair, no. This was just a hypothetical example.
Okay. And would those identify research needs or data gaps, as you call them?
Yes.
Okay.
Mr.
If
Chair.
you have specific items you believe need to have further research done on them, would you just put that in a list and somehow get it to us?
Yes,
And
Mr. Chair.
we have a research committee that'll be looking into those needs as well. And I notice a hand up of Dr. Carlson Van Dort. Doctor now, huh? Yeah.
No, I was going to say the same, Mr. Chair, thank you. And I would love to invite you to come chat with the research committee at some point in the relatively near future. Thank you.
Commissioner?
Yeah, I was talking to a few board members. The International Year of the Salmon has largely concluded its work, and now it's under analysis, and this is just a snapshot of what's coming our direction. The question is what happens next? And there's a lot of discussions internationally occurring as to what the next phase or development will be in the International Year of the Salmon. A lot of that has an added layer of complexity because of the Ukraine situation and inability to talk directly to Russia, for instance, between Alaska and Russia, but there are international dialogues ongoing to figure out what the next step in the International Year of the Salmon will be, and I'm convinced that there will be continued work in this area, but that work will be largely framed by what they find out with the ongoing analysis and largely the level of cooperation that occurs into the future. So it's not over yet.
Thank you. Any other questions? Mr. Carpenter.
Thank you, Mr. Chair. Dr. Howard, thanks for your presentation. I guess I'm referring to, I guess it's page 16. I'm not sure if I guess that's slide 16 also. you have some pie graphs there from 2019 to 22. And something that you said kind of caught my attention is, you know, when you look at these pie graphs and you think about, and you hear in all the reports that are being done and all the studies are being done and about how many pink salmon there are being put into the Gulf of Alaska, into the Bering Sea. And when this study was done, in some situations, obviously coho enumerated around the state are significantly less than there are pink salmon. And you hear about competition for food and things like that. Is there any speculation as to why more pink salmon weren't found in the areas that, you know, seem to have been reported that they would be in a lot of these published papers? Are they deeper? Are they, any speculation or any idea on that?
Through the chair, so from what we know about the depth distribution of pink salmon, they are relatively shallow. They should have been caught with the gear that we were using. Really, it's mostly Chinook that we were recognizing that we're unlikely to be caught proportional to how many are out there because they tend to swim quite deep compared to the other species. Nobody's quite sure what's happening with the pink salmon. I think the assumption is they must have been either further south than were sampled or further west than were sampled. We do know that some of the Asian pink salmon do overwinter in the Sea of Japan and the Sea of Okhotsk. So, you know, when we're talking about pink salmon overall, we know some of them aren't making it all the way into the North Pacific. They're staying a little more locally, but there should be pink salmon somewhere in the North Pacific. We just weren't encountering them in the numbers where we were looking, as was expected based on previous research in this region.
Thank you for that. So I guess my follow-up question would be, so if the research has shown in the past that there's likely to be some competition between the large levels of pink salmon that are being released into the Gulf of Alaska, and you look at these pie charts, when these surveys were done, and you found these higher populations of sockeye and chum, which you knew would probably be in those areas, but the pink salmon weren't in those areas, does that mean, or does that lead us to believe that either the areas that the pink salmon are overwintering are far different than we actually know, and does that actually suggest that maybe that the competition with these other species in these areas is actually less?
Through the chair, I think what it means is that we need to check our assumptions of what we think we know about salmon at sea. There are other times in the life history of these fish where, you know, we would expect them to overlap more with some of these other stocks and species, particularly at the juvenile life stage. When they first migrate out from their rivers, they tend to be a little bit more concentrated, local to their river system. And when these adults are returning to their river systems, they tend to be a little more concentrated to their river systems. But in winter, we don't have a lot of information about what these fish are doing in winter. And what we're learning is they're doing something different than what we assumed they were doing. So I think that's what we're learning from this research.
Thank you.
Okay. So all this is really exciting. This is cool stuff. I have two questions. And slide 8, if we could go there, I think it's slide 8. Go back one. Oh, darn. I'm looking at the one where it says chum salmon. There's no numbers on our papers here, so I don't really know where I'm. There. Okay.
Through the chair, that's 18.
18, okay. So my question is among so many other things that are somewhat surprising about that is I'm wondering about the high level of chums from that are considered from Japan and Russia, and in both areas, and especially compared to the Western Alaska, and considering the habitat that Japan has and Russia. It seems like an exceptionally large amount of fish being found in that area compared to Alaska. Are those all wild fish?
Through the chair, the vast majority, particularly from Japan, are hatchery-produced chum salmon.
Okay.
Although there are wild chum salmon, especially in the Bering Sea part of Russia's river systems. There aren't hatcheries in that portion of their distribution.
So if you go to slide, I guess, 19, and that pie chart there, there's a lot of pink, especially in the western side, central western or central North Pacific, which would account for those fish stocks as well, right? I guess that kind of surprises me, which would be an effect of hatcheries potentially. But now I'm going to ask my most layman term, or in layman's terms, but this is a layman question. Will you, I hear oftentimes about the idea of correlation versus causation, and that gets thrown back and forth a lot. And I think especially going for the next presentations we'll have throughout the day, can you kind of just explain to me causation or correlation versus how it leads to causation and vice versa? Because it's in order to talk to each other. I think you were hitting on that actually in some of your first slides, but would you explain that?
Through the chair. Yeah, so I think this slide I was attempting to give a hypothetical example of some of the pieces of information that might help us get to a conclusion that this correlation is potentially causative in nature. Because one of the challenges we have when we look, for example, at pink salmon abundance. Pink salmon abundance across the North Pacific has increased over time. That is true. The other thing that is true over that same period of time is that the temperature in the ocean has also increased. And so that's always been a really difficult thing to tease apart. And when you go to these kinds of meetings like the Northern Hemisphere pink salmon meeting, you get people, kind of struggling with this fact that we can't really pull apart the fact that the pink salmon abundance is also associated with temperature. So are things being driven by pink salmon, or are they being driven by temperature, or is it being driven by both? Those things are really tough to pull out with a lot of the kinds of information we have right now. And so that's why having some of these additional studies that can get at those pieces in between would be helpful to understand, okay, is this correlation indicative of a cause and effect, or is it something else?
Thank you. I mean, I feel like quite often when you live on your little stretch of river or whatever, like I do, I seem to, have blinders on what makes all this happen, or at least I try to figure it all out. And I think that's maybe human nature and what we deal with in today, but thank you.
Any other board questions on slides 18, 19, and 5 or 6, since that was with the correlation aspect of it? I do have a couple of questions, and we'll start with the earlier slides, 5 and 6. On slide 4, scientific literature. I am sure, knowing you by now, that you've read the September report that Dr. Ruggiero, is that how you pronounce his name, came out with. Have you read that report?
Yes, Mr. Chair.
Yeah, and it's been submitted by the Bering Sea Fishermen's Association as RC2. I'd highly encourage board members to not only read it, but to study it. It's an excellent report. But my question to you is that report sound science?
Yes, Mr. Chair. And I think they did I wish that report had come out before I made these slides, because they did a really good job of especially expressing this perspective of, you know, the people who are pretty convinced that that relationship has been established.
Yeah, I agree with you on timing. I wish it had come out earlier also, but nonetheless, it's out now, which I'm very happy about. On slide 4, that report seems like it would overlap number 2, 3, and 4 on slide 4. Am I reading that incorrectly?
That is a correct read, Mr. Chair. They did a good job of synthesizing the information that is already out there, and I believe they've referenced many of these papers in their report.
Yeah, and as the meeting goes on, I'll be referencing that report quite frequently. On the next slide, slide 5, if I heard you correctly, and you really need to correct me if I didn't, my understanding is you said the Asia reports dealt with it ecosystem-wide, whereas the reports that we do not do that because of a lack of data. Is that a correct statement of what you're trying to get across?
Mr. Chair, in general, that tends to be what we see, is that the Asian research is focused more on using survey data and those observations to inform their analyses of salmon competition at sea. And those just aren't generally available in the Eastern North Pacific to the same degree.
Okay, so if it were available, would their system of analyzing data, et cetera, and coming forward with conclusions be a better process?
Mr. Chair, I think it would address some of the issues that are brought up by the people who are not convinced that a cause-effect relationship has been established.
Okay. Now going to slides 18 and 19, which were covered by Mr. Wood previously. These and some of your other slides deal with catches and the analysis of what the catches are. Have you done an analysis of what the releases are from these various areas? In other words, how many hatchery fish are being released by Russia or by Japan or by Pacific Northwest, et cetera?
Mr. Chair, we have not. There is information out there on the releases that is published and available by the North Pacific Anadromous Fish Commission.
Would you send me the links to those, please?
Yes, sir.
Okay. Okay. Go ahead, Mr. Raybone.
That information will be presented in the next presentation, actually.
Okay, great. Okay, great. Okay, any other questions from board members on slides we haven't covered? Ms. Carlson-Van Dort.
Not on any slides in particular, but my question, Katie, is just whether or not there's a plan or the potential to be doing similar survey work in the Bering Sea with related, with respect to salmon species.
Through the chair, we do have an ongoing survey effort with our NOAA colleagues in the northern Bering Sea, and that's primarily focused on Yukon and Norton Sound stocks. We're working to develop a similar survey in the southern Bering Sea focused on Kuskokwim and Bristol Bay stocks. What we haven't done yet, but I think is the next step, is to do some of the kinds of analyses that the Russians have done with their survey work to look at, okay, what is the prey base that is available to these fish? And is it sufficient to support the number of fish that are out there? So that is a next step that hasn't been done yet.
Thank you. It's in my very humble and limited opinion that the work that you're doing and that the department is doing in this space is some of the most important scientific work on salmon that's occurring right now. And the information that will be provided, hopefully, is desperately needed, to try and understand more about these species. And to that effect, the surveying in the Gulf, the surveying the Bering, it sounds like you need a boat. And also, it also, you know, I think the importance of the collaborative work with other nations is also critical to try and supplement some of that. And I know that that requires international travel and things, Mr. Chairman, which I imagine is kind of expensive and whatnot. But I think that those are some things that we should be considering, at least weighing in on and putting on our wish list for you. Thank you.
And I totally agree with 100% of what was just put on the record by Ms. Carlson Van Dart. And I'll give you a little entree to sort of advocate for your efforts. You've asked for a new vessel to be used by the state and doing the research. And would that be addressing some of the issues you're describing in here?
Mr. Chair, yes, it would allow us to do some of the observations, the direct observations that are difficult to do right now.
Great. I was hoping maybe some movers and shakers dealing a budget could possibly be listening in and deal with that. I have a couple of questions on your analysis, and you see it on page 9 of slide 9, you see it elsewhere. You take pink salmon as a whole and tell us what the numbers are, but you don't break it down to wild versus hatchery. And I know that, for instance, Prince William Sound Science Center had done a weekly catch for several ongoing years. And what they found to be the case was they were catching 70, 80% that had hatchery genes or hatchery in their lineage somewhere, which means that the remainder would be wild. It would be interesting to me if you had 3 graphs, one showing the wild, one showing the hatchery, and one showing the total, because we may well be maintaining a level at the expense of the wild, but emphasizing the hatchery fish, and that would be helpful to me.
Yes, Mr. Chair. On a Pacific-wide look, not all countries record what their catches are, hatchery versus wild. So I'm not sure if we can get that information across all of this North Pacific region. They do report what is released.
Yeah. Thank you so very much. I'm really intrigued by what you're doing, and I highly encourage you to continue. And if you have any other needs, I'm sure the department would love to hear them, and I know our research committee certainly would. Commissioner.
Yeah, before we leave this talk, when I became commissioner, it became apparent to me that we needed to start looking in the ocean. So I tasked Bill Templin to actually put together a marine science program. And over the last 5 years, Dr. Howard's been hired and has put together a really good program. Bill's led that effort in terms of our international cooperation, and I want to take this opportunity to thank them. Our understanding of what's happening in the ocean is substantially increased, largely because of their efforts, and again, I want to thank them.
Any other questions of Dr. Howard? Thumbs up, you're right. Thank you, Dr. Howard. Does anybody need a break? Here in Sandoan, we'll go to the next presentation, which should be Lorna Wilson on the Alaska Fishery Enhancement. Is that correct?
And she is online.
Oh, she's online.
She was stuck in Juneau.
Having had that happen to me more than one occasion, I understand. Is it Dr. Wilson or Lorna Wilson, or do you know? I don't believe she's a doctor yet. Almost a doctor. Lorna, can you hear us?
Yes, can you hear me?
Yes, we can. You have the floor.
Great. Let's see. Oh, I think we need to have, oh great, are you stopping the Katie's presentation? Okay.
We cannot see you, so we rely entirely, there we go. We got somebody in control of the slides, I guess. No, we don't. That's the slide for another one.
She's got to patch it over to Zoom.
Do you want to take a break?
No, I think this will only take us.
Okay.
Savannah, would you like a break to do this transition? Okay, yeah.
Take a couple of minute break until we're ready to proceed technically.
It says you cannot start. Oh, there we go. Great. I think I can share my screen now. fishery enhancement program is stakeholder-driven. A link with information on the next regional planning team meeting in each area is in the bottom of the slide. So the RPT meets annually or as necessary in order to fulfill its advisory role to the commissioner on regional salmon fishery enhancement activities. They draft and update the regional salmon plan and provide those to the commissioner for approval. They review hatchery permit applications and provide recommendations to the commissioner. And then following those substantial ongoing work, they comment on proposed P&P permit suspension or revocation. And in Southeast, they provide annual recommendations to the commissioner on production changes. And the RPT may provide support or review of regional fisheries enhancement in other areas at the commissioner's discretion. So the RPT's main responsibility is preparation of the Regional Comprehensive Salmon Plan, or CSP. The CSP describes rehabilitation of natural stocks and supplementation of natural production in the region. It considers the needs of all user groups and defines production goals by species, area, and time. It should include provisions for both public and private nonprofit hatchery systems. The RPT drafts the comprehensive salmon plan. The plan gets public review. The RPT considers those public comments and revises the CSP. This plan is then sent to the commissioner, who may ask the RPT to revise the CSP or may approve the CSP. The approved CSPs and those out for review are at the link at the bottom of the slide. Only private nonprofit corporations, or P&Ps, can operate a hatchery under a P&P hatchery permit. Oversight of P&Ps is by the Nonprofit Corporation Act. P&Ps have a board of directors with at least one representative of each user group that sets production goals and oversees business operations. The board of directors usually forms committees, such as the production planning and finance committees, that make detailed recommendations to their board. A P&P corporation can be regional, like a regional aquaculture association, or it can be non-regional. The first step in a P&P hatchery application process is a management feasibility analysis, which basically reviews whether the hatchery fits in regional fisheries management. Questions are if the hatchery will contribute to the common property fishery, is how big and what is the location of the special harvest area, are there special management considerations like nearby stocks of concern, are there fish available for broodstock, and can the hatchery produce a number of fish? The application is reviewed by the regional planning team, reviewing many of the same things as a MFA, or management feasibility analysis, along with compatibility with a comprehensive salmon plan. The P&P hatchery application gets a public hearing that's publicly noticed, and then P&P permits are approved by the commissioner based on the RPT review, department review, and the public hearing, with their decision based on 8 criteria. Basically, will the hatchery work? Those 8 criteria are in regulation. So a P&P permit authorizes P&P hatchery operation. The permit specifies the species, egg source, egg capacity, release locations, and other conditions. They typically contain the condition that the operator is required to remove unharvested fish in the terminal area. P&P permits do not expire, but they can be relinquished by the permit holder or revoked by the commissioner. I will show you permitted egg capacities later in this presentation. Fish transport permits are required to collect, transport, possess, propagate, export from the state, or release into the waters or lands of the state any aquatic organism. Fish transport permits are specific to the hatchery, as species, and stock. They specify the maximum egg number of fish and locations, for example. P&P hatchery fish transport permits must comply with a P&P hatchery permit. Fish transport permit applications are reviewed by regional sport and commercial managers, genetic staff, and pathology staff. FTPs are approved by the commissioner. A few years ago, FTPs were put in a database and online, which is very handy for operators and Fish and Game reviewers and their coordination. Terminal areas are designated by the Commissioner, Board of Fisheries regulation, or by emergency order for fisheries enhancement, and are areas where hatchery returns have a reasonable degree of segregation from naturally occurring stocks and may be harvested in the common property fishery without adverse effects. A special harvest area, or SHA, is the type of terminal harvest area where hatchery returns are to be harvested by the hatchery operator, and in some situations by the common property fishery. SHAs may be designated in the P&P permit also. The Alaska statute also provides for a hatchery permit holder to conduct a cost recovery fishery through a common property fishery called a fishery assessment. In a fishery assessment, the commercial fleet harvest of hatchery returns in the terminal area is taxed. The tax is collected by the processor, submitted to the Department of Revenue, routed through the state, and provided back to the P&B Corporation. This program was first used in 2012 for the Hidden Falls Hatchery terminal fishery in southeast Alaska. These pictures show the same fleet fishing at Hidden Falls terminal harvest area. A funding source for regional aquaculture associations specifically is the Salmon Enhancement Tax, or SET. Commercial salmon fishing permit holders vote to self-impose SET on the sale of salmon in their region. These funds are collected by the state and distributed to the RAA to finance hatchery operations or other enhancement and rehabilitation activities. Non-regional P&P corporations do not receive SET funds. The plot shows the SET collected statewide in the last 3 fiscal years. Hatchery production planning is a continuous process. In the fall, hatcheries have a brood goal, which is the number of fish at the hatchery needed to achieve an egg goal. After eggs are taken, juveniles are reared at the hatchery over winter and up to 2 years before they are released at permitted sites. The hatchery rearing depends on species life history. Hatcheries then project these fish will return 1 to 4 years later, again depending on the species life history, with fish contributing to common property fisheries, cost recovery fisheries, and broodstock for egg takes, allowing future production. The salmon life cycle considerations mean that some species are more economical to rear. Pink salmon have the shortest rearing time at the hatchery and spend 1 year at sea for the highest and fastest return on investment. Chum salmon have the same rearing time at the hatchery, but have a longer life cycle. They spend 3 to 4 years at sea, so they have a high but longer return on investment. Chinook, sockeye, and koa salmon have longer rearing time in freshwater, typically 2 years, but koa will return after 1 year at sea, but Chinook and sockeye have a long return on investment, typically spending 2 to 4 years at sea. So pink and chum salmon are the bulk of Alaska hatchery production because they have the highest return on investment for their cost of production. For a hatchery operator to expand production, add a species or stock, or move production to a new site, they need to submit a permit alteration request, or PAR, back to the P&P coordinator. PARs are due February 15th, but the deadline can be extended if justified by extraordinary circumstances or emergency. PARs are reviewed by fisheries management, pathology, genetics, and aquaculture sections. A lot of the time PARs are reviewed by regional planning teams, usually in the spring meeting, and then a decision on permit alteration on PAR approval is typically in April or May. If a PAR is approved, the P&P permit is then altered. Alaska hatcheries mark their releases primarily to estimate their contribution to fisheries. The otoliths, or fish inner ear bones, are thermal marked by changing the rearing water temperature, moisture, or sometimes salt water at set cycles, leaving bands that can be seen under a microscope. Using otolith data, catch can be apportioned to origin by looking for these thermal marks. 100% of the release can be thermal marked, so otolith sampling can provide data with very, very high confidence. Much of otolith mark recovery is performed by hatcheries, by hatchery operators. Some releases, particularly Chinook and coho salmon in Southeast, are marked by inserting a coded wire tag, a small piece of metal wire etched with a code, into the nose of the fish. Tagged fish also have their adipose fin, a small fin on their back, clipped off so tagged fish can be visually inspected in the return. Tags are applied to only a portion of the release, which adds uncertainty to apportionment estimates. Each year, hatchery operators and ADF&G communicate their plans for the season in annual management plans, or AMPs. AMPs organize and guide the hatchery's operations regarding production goals, broodstock development, and harvest management of hatchery returns. They typically contain things like the current year's egg take goals, the planned releases of juvenile salmon, the remaining fish inventory for release the following year, and the expected returns that year by location. The harvest management plans of returns, and the P&P and fish transport permits allowing these activities. They sometimes reference evaluation plans, like their marking and sampling plans, and the current year's AMPs are available online at the link. The process of developing the AMPs is that the operator drafts the AMP, submits to the department, we review and edit, submit the draft back to the operator, they review our edits, and they send it back to us, back and forth until we have a final draft that everyone's happy with, and then we send our plans to the commissioner for approval. Hatchery annual reports are reported by stock and species. They include things like the broodstock source and numbers, the number of eggs taken at a hatchery, the number of juvenile salmon released, and the adult returns attributable to the hatchery in cost recovery harvest, their contribution to common property fisheries, so commercial sport, personal use, and subsistence, the escapement if required, the number of fish used for brood, and typically it can contain forecasted returns. Annual reports are due each year by December 15th. Hatchery annual report data provides department oversight to ensure production is as permitted. Annual report data is used in season summaries, annual fishery management reports, projections, harvest projections for the subsequent year, and our section prepares a comprehensive annual fisheries enhancement report for the legislature. A link to the 2022 report is in the middle of the slide and provided in today's meeting documents. We can provide reports to the Department of Commerce as P&P production may pertain to loans provided through the Fisheries Enhancement Loan Program. Other interested data users are economic groups like the McKinley Research Group and researchers who may be looking at correlations with hatchery production or genetic studies. Next, I will show trends in enhanced production of salmon, starting with the North Pacific. And then for Alaska, I will start by showing the number of eggs taken at hatcheries and permitted capacities by species. And then I will show production by area and maps, and then production for statewide and then by region. Here's the production of salmon by hatcheries in the North Pacific, 1952 through 2022, with colors by species. Chum salmon, the darker blue, second color from the bottom, is by far the predominant species produced, followed by pink salmon, the light blue on the bottom. The number of salmon released into the North Pacific has been fairly stable since the 1990s. This plot was provided by the North Pacific Anadromous Fish Commission website. Here's the same production data by country of origin, with the U.S. broken into Alaska, green, the third from the bottom, and the rest of the U.S., dark blue on the bottom. With each bar is showing the total number of fish released, 1952 through 2022. Here are locations of the 27 hatcheries in Alaska that released fish in 2022. Alaska's production is out of Southeast, Coastal, South Central, and Kodiak. In the following plots, the bars are the number of eggs. The darker shades are eggs taken by P&P hatcheries, while the lighter shades are eggs taken by state hatcheries, so Fisheries Rehabilitation Enhancement and Development, FRED, and sport fish hatcheries. The black line is P&P hatcheries' permanent capacities. These are minimum permanent capacities, as capacity from 4 hatcheries that do not produce fish are not included. Statewide, the number of Chinook salmon eggs taken has been fairly stable, under 20 million. The P&P permanent capacity of Chinook salmon eggs has been continuously increasing, while P&P hatchery egg takes have only slightly increased. Hatcheries can often only rear a portion of their Chinook salmon egg capacity because they are expensive to rear and take a long time to return. This is the P&P permanent capacity of sockeye salmon has remained stable at 160 million eggs since 2001. Sockeye egg takes have declined statewide since the 1990s. Some P&P hatcheries that have permanent capacity are not using the capacity because of the considerable investment needed for a successful sockeye program. Among many things, the water source needs to be virus-free, and sockeye need to be reared separately from chum and Chinook salmon, which means that facilities releasing sockeye typically only produce sockeye. And sockeye egg takes have declined in every area, although declines are not as severe in Southeast, where there are federal funds that support the program. The P&P permanent capacity of coho salmon has been steadily increasing, and so has the eggs taken by P&P hatcheries. Some coho production is to provide an alternative to reduce Chinook salmon availability. The P&P permanent capacity of pink salmon has remained stable, between 1.3 and 1.4 billion eggs since around 1992. P&P hatcheries' permanent pink salmon capacity is 40.5 million eggs lower today than in 1994 through 1996. Chum salmon permanent capacity has been variable over time. It declined in the 1990s and has been steadily increasing since then. Here is the percent of permanent egg capacity used by P&B hatcheries in 2022. Much of the chum salmon permitted egg capacity, so the white dotted, was taken by P&B hatcheries in Southeast, Prince William Sound, and Kodiak. Much of the pink salmon permitted capacity, so the pink dotted, was taken by P&B hatcheries in Prince William Sound and Kodiak. Production numbers, so the number of fish released, are limited by the number of eggs taken, which means that unless permitted P&B hatchery capacity increases in areas where hatcheries are taking their permitted capacity, the production will be fairly stable into the future. The number of eggs taken in all hatcheries, including P&P hatcheries by species, is in Appendix G1 of the annual Fisheries Enhancement Report, and shown in this table. The permitted capacity of PNP hatcheries by species is in Appendix B1 of the Annual Fisheries Enhancement Report and is shown in this table. Here is the production statewide of salmon by Alaska hatcheries, 1975 through 2022. Pink salmon, the pink dotted, are the predominant species produced, followed by chum salmon, the white dotted. Statewide, Alaska hatchery production has been fairly stable since the 1990s, mid-1990s. Here is the same data broken into mark type. In the 1970s and early 1980s, most production was not marked, the white with dots. Hatcheries have been increasingly using coded wire tags, the black, and otolith marks, green with dots, to mark releases. In 2022, 95% of Alaska's production was marked. Southern Southeast has 6 hatcheries operated under PNP permits by the Regional Aquaculture Association. One of these is state-owned. Also, there's 1 hatchery operated by a PNP under a cooperative agreement with ADF&G Sportfish, and 1 hatchery operated by Metlakatla Indian Community, which is sovereign and governs their own hatchery and fisheries. Northern Southeast has 8 hatcheries operated under PNP permits, 4 operated by the Regional Aquaculture Association, and 4 operated by non-regional PNPs. One of these is state-owned. There is 1 federal research facility. In 2023, all PNP hatchery permit alteration requests and applications in Alaska were in the northern southeast planning area. The Hidden Falls Hatchery PNP permit was altered to allow up to 40 million chum salmon green eggs to be taken at Hidden Falls Hatchery on behalf of the Port Armstrong Hatchery PNP permit for release for release at Hidden Falls Hatchery permitted release sites. There is no overall change in the permitted egg capacity. And then also, Northern Southeast Regional Aquaculture Association started the application process for a PNP permit to operate Little Port Walter Hatchery. Here is the production of salmon by Southeast hatcheries, 1975 through 2022. Most of the production is of chum. Production of chum salmon increased dramatically during the 1980s. Since then, since the 1990s, increases have slowed. Chum salmon contribute to the common property seine, gillnet, and troll fisheries. Chum and pink salmon are the primary cost recovery species, paying for the operation and production of chum, chinook, coho, and sockeye salmon. In Prince William Sound Copper River area, 5 PNP hatcheries are operated by the Regional Aquaculture Association, and 3 of these are state-owned. 1 hatchery is operated by a non-regional PNP. Here is the production of salmon by Prince William Sound Copper River hatcheries, 1975 through 2022. Pink salmon are the predominant species produced, followed by chum and sockeye salmon, contributing significantly to the common property seine and gillnet fisheries. Pink salmon are the primary cost recovery fisheries species, paying for operations and the production of sockeye and coho salmon. Cook Inlet has 3 hatcheries operated by the Regional Aquaculture Association. Of these, 2 are state-owned and under contract with ADF&G. 1 hatchery is operated by the state. There are 2 hatcheries in Kodiak that are operated by the Regional Aquaculture Association. Both are state-owned under contract with ADF&G. There's 1 hatchery in interior Alaska and Fairbanks, operated by the state. Here is the production of salmon by Cook Inlet hatcheries, 1975 through 2022. Pink salmon production was suspended for a few years in the 2000s due to economic reasons, but has resumed and the programs are rebuilding. Sockeye salmon production contributes to seine and sport fisheries. Cost recovery occurs by harvesting a portion of fish returning to the hatchery's release sites and is a mix of sockeye and pink salmon, which is used to pay for the production of all species and non-propagated fisheries enhancement. ADF&G Sportfish Division operates the William Jack Hernandez Sportfish Hatchery in Anchorage. They stock lakes and release Chinook and coho salmon and other species to benefit sport fisheries. Here's the production of salmon by Kodiak Hatcheries, 1975 through 2022. Kodiak Hatcheries produce predominantly pink salmon, contributing significantly to the common property seine fisheries, as well as the primary cost recovery species paying for operations and production of chum, sockeye, and coho salmon. Before I talk about returns, I will walk through a rough indice of survival using pink salmon numbers because they have the simplest life history. The column to the far right, green, is the percent of release to return. It's like marine survival. This is found by dividing the number of attributable returns, 28 million, reported by hatcheries, by the number of fish released, 870 million. The egg to release percentage, column blue, is the number of fish released, 870 million, divided by the number of eggs taken, 982 million. Alaska salmon hatcheries produce salmon with good survivals. Freshwater survival in hatcheries is higher for all species than for wild or naturally reared fish in blue font. Marine survivals are comparable between hatchery and wild. Are comparable between hatchery and wild reared fish, although returns of pink salmon and chum salmon, for which there's extensive sampling of returns, is better known. Returns of coho, chinook, and sockeye salmon are known to be underestimated because their contributions are not always estimated. For example, hatchery contribution to sport harvest was not estimated at Cookturn and Nilchik, which are designated sport projects. Returns of all species are likely to be underestimated because not all mixed stock fisheries are sampled for stock composition. Alaska's production has translated into fisheries enhancement. Here is a statewide contribution to commercial fisheries. Common property is the blue shaded, and cost recovery fisheries is the white with dots, 1977 through 2022. The solid black line is the percent of hatchery-produced fish in the total commercial harvest, in Alaska's total commercial harvest. In 2022, hatchery-produced fish were 25% of the total commercial harvest, just under the 10-year average, the dashed line, of 30% of the total commercial harvest. Here's Alaska's commercial salmon harvest from 1900 through 2022. The pale yellow is commercial wild stock harvest, and the dark blue is commercial hatchery harvest. Alaska salmon fisheries enhancement planning and fisheries management allowed for harvest and supplementation, yielding both high wild and hatchery stock harvest. So here's a slide showing some recent concerns and research happening by Alaska's hatcheries. So and ADF&G. So there's the Alaska Hatchery Research Project, which is ongoing, and we'll see some presentations on this later this morning. There's ocean ecology. We're wondering what's going on with salmon at sea, and ADF&G, we hired our first scientist on salmon ocean ecology in 2020. In Southeast Alaska, hatcheries have suspected that predators of released salmon, like whales and sablefish, may be reducing their marine survival. And so in response, some hatcheries are towing net pens or transporting fish in a vessel or other post-molt juvenile salmon offshore before release to avoid nearshore predators for the purpose of increasing survival. But because of concern that transport can impact homing, their fish transport permits were approved with conditions such as maximum distance and requiring evaluation of impacts of the transport on homing behavior. In Southeast, hatcheries are started using tagging trailers to increase the tag rates of Chinook salmon to increase data confidence. NSERA, Northern Southeast Regional Aquaculture Association, has sampled nearshore fish species composition, biomass, and isotopes to investigate the cause to help better understand cause of declining marine survival. NSERA is starting to look at growth measurements to better understand some times high proportions of age 3, so younger maturing chum salmon, which can complicate their forecasting of returns. They're wondering if the early life growth shown on scales can help predict the age shifts. There's cooperative work between NSERA, Sitka Sound Science Center, NOAA, and the university looking at if amino acids derived from kelp and red algae can help improve homing efficiency of chum salmon. And then hatcheries in Southeast and Prince William Sound are looking at phytoplankton bloom timing to see if that can predict zooplankton bloom timing to optimize release timing. In southern Southeast, their hatcheries are looking at if developing their understanding of the currents, nutrients, seasonality, and temperature and salinity for potential aquaculture at salmon release sites. In Cook Inlet area, there's reduced non-hatchery fishery enhancement. So projects at like fertilization at Leisure Lake and Bear Lake and Delight Lake weir operations were suspended for budgetary reasons. But there are other non-hatchery fishery enhancement projects, pike eradication, beaver dam monitoring, flow control monitoring to provide water for fish in spawning areas, so non-hatchery propagation, and culvert monitoring are ongoing. And then predators are being observed, like mammals, sea lions have been observed in increasing numbers in terminal areas. So for example, in 2022, sea lions consumed an estimated 8,800 pounds of fish per day at Wally Nirenberg Hatchery. So they're trying things like exclusion devices, putting a cone on the buoys in the area to prevent sea lions from hauling out in the nearby area, and also acoustic seal fences being trialed to protect broodstock. These are some of the recent concerns and research ongoing at Alaska hatcheries. Thank you. This concludes my presentation. I'd like to thank you for this opportunity to talk about Alaska's salmon fishery enhancement program.
Yeah, thank you. There are very few prerogatives of the chair or advantages, but one of which is to schedule the meeting progress, and we're going to take a 15-minute break and then get to questions from staff. You'd like to add one thing, okay?
Yeah, I just wanted to add one thing to the presentation. When I became commissioner, I capped the egg capacity as a policy on pinks and chums, not the number of releases, but the egg capacity in pinks and chums until we had some answers as to these hatchery wild interactions. And that wasn't mentioned in it, but that's a policy that I've directed at the department level.
Doesn't egg take and release ought to go hand in hand?
Egg capacity. So that you may have increased survival within the hatchery, so you have higher releases, but the egg capacity, the egg take is what I've capped.
Thank you. We'll just take a 15-minute break, come back to question. We're going to start off, and Mr. Godfrey will be rejoining us shortly. So we're at the point where the questions of the presentation that was just made by Mr. Wilson. Any questions from board members? Mr. Swenson.
I guess now I'm going to, through the chair to Sam, my understanding was that the hatchery fry, when they go out, would be bigger because they're kept in pens than the natural ones that go out smaller. And I think that Sam can speak to this, and I think I'd like to have him speak to that.
Ray Bung.
Yeah, thank you, through the chair. So when the fry hit the salt water, when they leave fresh water, so we're talking about pinks here, or chums, they're the same size as their naturally spawned counterparts, the wild fish. They hit salt water at the same size. The difference is that in Alaska, hatchery programs have evolved to, instead of direct release of what we call unfed fry, they go into net pens, and they're short-term net pen reared for 2 to 3 months prior to being released. And what it accomplishes a couple things. First of all, when you release unfed fry, they're going to rear near shore for 2 to 3 months until they get to a size large enough to head offshore, same as the wild fish that are doing that. So there'd be near shore competition. NOAA's work, and I think Dr. Katie Howard was involved with that with some other species, is that the majority of mortality occurs in those first couple months in the ocean. After that, it kind of, that's about the number that's going to make it, roughly. So when they're released from the net pens, they're quite a bit larger. In the case of pink salmon, they're double to maybe triple the size, depending on the location and the temperatures, as they were when they entered saltwater. But at the same time, their wild cohorts are also that same size because they've been rearing naturally near shore and growing. They need to reach what we call a size threshold before they can go offshore. In Southeast, the SEACUM study, the Southeast Coastal Monitoring, has been sampling these fish for decades now and monitoring that. And it's a great forecasting tool for our pink salmon, for example, because once they're heading offshore, that number correlates really well with what's going to come back as adults. But back to your original question, they enter saltwater at the same size. They're held in net pens and fed by the hatcheries to get them up to size to go offshore, while the wild cohort are feeding near shore and growing to that same size to head offshore. So they're approximately equal in size when they leave the near shore areas. Does that answer it?
Yes, but does that differentiate by location at all, or are they all the same in that respect, whether it's Southeast, Prince William Sound, wherever? Does that always follow the same pattern?
Well, the one thing I've learned about fish is that as soon as you think you know something about fish, they'll prove you wrong. There's a lot of variability, but in general, yes, it's the same pattern. Prince William Sound net pen rears their fish to get them to a size to head offshore, and they head offshore pretty quickly. It's pretty much a beeline.
Do they do that in the hatchery with all species of salmon?
No. So if it's a marine release, then yes, I would say I can't think of a program off the top of my head that doesn't do short-term net pen rearing in saltwater for at least 2 to 3 months. So that would be pinks, chums, coho, chinook. But there are freshwater releases, for example, unfed sockeye fry, or sometimes there's some programs that we have that stock lakes with unfed fry, and that's limited by the carrying capacity of the lake through freshwater plankton studies and things like that. That tells us how many that lake can support. And there are some freshwater releases of chinook and coho, I believe.
So does that, if the freshwater releases hatchery fish that go out are bigger than the natural ones, over time, what is that going to do? How is that going to affect between the two runs?
Let me clarify, if it's a freshwater release, they're the same size as their wild conscripts when they're put into the cohorts, when they're put into the fresh water. That, we call that like Golcana Hatchery stocks the lakes up there that feed into the copper, and those are unfed fry, so they swim out of the gravel, they go right into the lake, the same as the wild fish that have swum out of the gravel. And in some cases, they may be even a little bit smaller than the wild ones that may have swum up earlier and started feeding already. So it varies based on temperature and things like that.
Okay, great. Well, that's good information. Thank you.
Any other questions?
Thank you. I've got some questions, and it's kind of to build off some of the commissioners' comments before we took a break. And also, I will just preface my questions that I was just sort of glancing through the titles of the coming presentations, and I haven't had a chance to preview them yet. So if any of my questions are better addressed or will be addressed with more specificity, and those, just let me know. But I guess some of my questions were around the permits, and I'm thinking about changes in production and the permit alteration requests and the regional planning team process and all those things. How are market considerations taken into account? So is there flexibility to produce, I mean, obviously there's flexibility to produce below the permit threshold. I'm assuming there's a process by which the commissioner reviews and reduces permitted production, I'm assuming, based on his just statements. But I'm just kind of wondering how the market considerations are taken into account on an annual basis, if at all, just considering some of the things that we saw in the last year and the tremendous backlog of product, I think, from 2-3 years that has been sitting on there. How does that affect production levels under the permit by, sort of individual hatcheries? Sam, please.
Thank you. Through the chair, there's probably a lot of people in this room right now, including Mr. Carpenter, who could probably explain this better than me, but I'm just going to speak to the PNP model. The PNP organizations are made up of permit holders primarily, so the fishermen that harvest the fish. That is the feedback mechanism. They drive what production they want to put in the water because it affects their take-home pay. So if prices are very high, of course they're going to want to increase and maximize production. If prices are low, they're going to consider whether or not it's a good return on investment. But the feedback mechanism is the fleet itself, the permit holders themselves, the commercial fishery permit holders themselves.
Thank you. If I might, with respect to kind of the slides that we had, 34 through, 38 or so, where it just delineated the PNP permitted capacity. In some of these species where the permitted capacity is considerably higher than has been utilized for many years, it looks like. I mean, should that capacity be reduced? I mean, just to clean it up on the books,
And might I add, Lorna, if you want to jump in, we can't see you, so please speak up if you want to respond to one of these questions. Mr. Raybon.
I'll go ahead.
Okay.
Yeah. So for the pink and chums, they're primarily at capacity. If they're below permitted capacity, it's generally because there was insufficient, when you look at the release number, it's generally because there was insufficient broodstock that year, or they had poor egg survival, or both. So I will point out, if you look at the chum graph, the drop in the late 90s in permitted capacity, that was a voluntary decrease of latent, we called it latent permitted capacity, where the hatchery operators had more permitted capacity than they had the capacity to produce, and so they voluntarily relinquished paper fish, if you will, that they didn't have the capacity to produce anyway, with the understanding that if they ever wanted to do more, they'd have to go through the process to seek a permit alteration request, et cetera. The other species, So the rearing fish, we call them Chinook and coho primarily. Those are constrained by, of course, broodstock and egg survival, but they're also constrained by water. They take fresh water for at least a year, and if you don't have enough fresh water or enough raceways or round ponds or whatever you're using, you can't achieve that. But sometimes they mix and match between those rearing species. Chinook and coho are sometimes referred to as Chinook equivalents because they have the same freshwater life cycle, and they are targeted by similar user groups. And Chinook are very expensive to rear, and frankly, they're wimps. They don't survive as well as some of the other species. Coho do a much better job. And coho are a much quicker return on investment. So that is another business decision that PNPs often will make, is the trade-off between the two. And that's also driven by price, but there is a bigger lag time with those species to respond to pricing. Again, Mr. Carpenter or several people in this room could probably explain that a lot better than I can.
Yeah, I guess what I was getting at, and thank you for that, Sam, is just like, there's a pretty big delta between what the state and the PNPs are producing and what the permitted capacity is, particularly for Chinook and sockeye, more so for Chinook, obviously. And I'm just, if that capacity should be reduced since it's not being utilized for some reason.
Yeah, if I may, one other thing I forgot to mention is that, again, Chinook are expensive to rear, and in general, they have to be paid for by something else. They don't contribute enough cost recovery to pay for their own production. So if you're struggling to make ends meet with cost recovery or salmon enhancement tax, or in some cases, funds from Sport Fish Division to contract that production out, if there's not enough, then that Chinook production will be decreased.
If I might, if under the permit, can you like bank eggs? Is that a common practice where you basically store eggs from year to year, and then when it's, or is it you take and then you use what you take the next year? How does that work?
So it's an annual egg take limit, the egg number. You can't bank them or roll them over or anything. It's a cap every year.
Which kind of got to my, the commissioner's question, because I was trying to figure out, like, can you release more than is permitted by take? So the permits, on one of the slides, it stipulated that the permits control or will stipulate the release locations, but what about the release amounts? I'm assuming that would be a function of egg take, but.
So the egg take is the ultimate ceiling on what can be produced. That's your first stopping point. We do have some, what we call remote release sites, where we have, limited the amount that can, of fish that could be a release there as well. So the egg take can either be released on site at the hatchery, and that's the simplest form, or a portion of the eggs taken at the hatchery that become fry or smolt can be released at various terminal harvest areas. Oftentimes the terminal harvest areas have a cap on the number of fish that can be released at them, but they all have to fall within the permitted capacity of eggs. You can't exceed that permitted capacity of eggs. So you just divide the pie amongst your release sites, kind of.
And thank you for that. My last question is kind of related to slides 52 and 53, and I'm probably not reading these very well, and I'm sure the information is embedded in there somewhere. But so what I was kind of interested in taking a look at were those releases. So what the release contributions are to commercial fisheries. So I see the egg take year, the release year, number of fish released. I would be really interested in seeing a graphic like the one on 53 for each of the hatcheries to just kind of get an idea of what their egg take was, what it was released, and then what those annual contributions were to both the commercial and the cost recovery fisheries.
Well, that's an easy one. That's reported every year in the annual reports, and we compile them into a statewide annual report that's provided to the legislature, and we provided to you guys in the past. She has links to that report on this page. We put out a new one every year. I think she's scrolling to the,
I figured it was in there somewhere, so we're
Yeah.
going to see it somewhere. Right?
there's tables, lots of tables in those reports, and all of those things that you're asking for are in there.
Thank you.
You just got to find it. You just got to find it. Any other questions? You know, I really encourage board members, if there's anything you want to know about hatcheries, this is a meeting to find out, so don't be bashful about asking. Mrs. Ray, you're trying to get my attention.
Yes, thank you, Mr. Chair. This would go to the presentation we just have, and if Lorna could maybe talk to this. Anyway, this is an incredible amount of information. I'm learning a lot here that I never really knew about. But anyway, on slide 20, on the terminal harvest areas, it was just something that I have a lot of questions I could ask, but I'm only going to ask one. Under the terminal harvest areas where hatchery returns have achieved a reasonable degree of segregation from natural occurring stocks, under that section there, if you could just maybe speak to overall how well you think this is working, like maybe like an opinion, you know, here in Alaska, and then if you could kind of give an example of a hatchery situation that has achieved a good degree of segregation and one that has not. And if you don't have something, you know, an actual situation that you can refer to, just an example, a theoretical example of how this would not be working. I'm trying to understand this whole thing.
Yeah, you have several
Thank you.
questions in there. Lorna, do you understand the questions, and can you respond to them?
I believe I do understand the question.
Okay. Proceed. Yeah.
So terminal harvest areas are often sampled by hatchery operators to know the composition of the return. And so they'll sample odorless and tags from the terminal harvest area to get, like, for example, the age composition or of fish that are harvested in order to better understand their stocks and their performance. And those samples indicate any fish that are not marked, or an estimate of the fish that are not marked would indicate non-hatchery fish in the terminal area. And every time I've reviewed that data, all fish, effectively all fish in the terminal area are very, like, 99% or higher on average are hatchery origin fish. So if a terminal harvest area is located, like, next to stock of concern, for example, that would be not an effective terminal harvest area location. And so terminal harvest areas and special harvest areas are reviewed by fishery managers for providing information before the area is designated as a terminal area. And so we have oversight over those locations. Does that help?
Yes, through the chair. Thank you. That helps immensely. And part of what made me ask this question is, you know, you hear things all the time as a board member. People tell me things and stuff, and it's, you know, I did hear of a situation where it was, you know, the percent was like 40% wild stocks mixed in with, and so it's interesting, and it clears up a lot to hear you talk about and give, that higher percent of hatchery fish versus wild fish. So thank you very much. Yeah.
Yep. There are sometimes non-terminal areas are often, sorry, not sometimes, often non-terminal areas next to the terminal areas are also sampled. And so maybe the 40% are fish that are intercepted in an area next to the terminal harvest area. That makes sense for fish that are a group of fish that are migrating through to their ultimate destination, the hatchery or wild stock. Their locations between the open ocean and the hatchery, some are mixed stock until they get basically to the terminal area. And so it could be that that 40% is a non-THA or SHA area.
Is that generated a question, Mrs. Roy?
Just a statement.
Go for it.
Yeah. It was expressed as a terminal harvest area, but, I realize that, non-terminal harvest areas, where they of course mix. And thank you very much for that.
Yep.
Okay. Any other questions? Okay, the chair does have a series of questions on different slides. I'll start at the beginning. Slide number 5. What evidence do you use to determine that the program is operated without adversely affecting natural stocks of fish with each of the hatcheries that are permitted? Lorna?
So this is not my decision. I'm part of a group of people that are following this act. One program that we've initiated has been initiated in Alaska is the Alaska Hatchery Research Project. There's a concern that strays entering natural streams could be adversely affecting natural stocks. Okay. And so that's an area of evaluation you'll be hearing an update on in an upcoming.
Yeah, I appreciate that. The statute's been in existence since 1974. The program you just described is relatively recent, within the last couple of years. There must be some program or process that the department goes and says, an evaluation process that says these programs and each and every one of these hatcheries is being operated without adversely affecting natural stocks of fish. Yeah, Mr. Raybung.
Thank you, Mr. Chair. Prior to initiating specific studies to work on these pieces of that, what we looked at a lot was the graph on slide 54, I believe it is, showing the commercial harvest of all stocks. And what you'll see there is that natural productivity, natural harvest hasn't really decreased since before the PNP program existed. Another thing we look at is, are we achieving the escapement goals in systems around the programs, the projects? Those are all indicators that natural productivity is doing okay. But that wasn't enough, so that's why we've initiated the hatchery research projects.
So if they're meeting escapement goals, that would be one of the tests, if I heard you correctly. But that doesn't differentiate between wild and fish and the hatchery, so.
And so we've sampled some of those systems for otoliths and things like that to see what those proportions are. And so those have all, each time we analyze something, it leads us to another rabbit trail to go down and dig into more. It's a never-ending, or I would say evolving, evaluation.
That's part of why you have your job. Next is still on slide 5, and it deals with the same statute, and it dictates allowing reasonable segregation of the returning hatchery. And I'll give you a hypothetical rather than to get into actual scenario. Let's assume that a hatchery releases 100,000 fish, and only 10,000 are returning because of mortality out in the marine environment. Then they go through the harvest area for cost recovery, and out of the 100 that came back, or 1,000, whatever the case may be, say 1,000 of those go to cost recovery. Then you go to the terminal harvest area, and they're harvested, and another 50. Remaining, there's 40 fish out there somewhere that haven't been harvested. Now, they have no natal stream because they were not raised that way. What becomes of those fish? It seems like they will be commingling with wild stocks, will they not? Mr. Raybon.
Thank you, Mr. Chair. Yeah, they will. So our policy and our practice is to require that those fish be taken out of the water to the practical extent possible. It's kind of hard to find 40 fish sometimes. But our goal is to have those fish harvested. The Alaska Hatchery Research Project did some really cool work in Prince William Sound. They did 3 years in a row of ocean entrance sampling and did a full accounting of the returns and demonstrated that about 95% of the hatchery produced fish were harvested at the same time that around 50%, if I remember my numbers right, 45, 50% of the wild stocks were harvested. So it showed that the management strategies that we have in place in that area, that specific system, are very effective by design to get those fish harvested. So the goal is to get them harvested. That said, we knew in the 70s that salmon stray. If it weren't for straying, we wouldn't have salmon in Alaska. This was all under ice 10,000 years ago. It's a survival mechanism. So we anticipated that. I say we, I really wasn't directly involved. But, and that's why we have the genetic policy. We require the use of local stocks because if they're going to stray into local stocks. So we want them to be similar, same genotype as where they're straying into, so they're not making a significant genetic effect on any local stocks. That was built into the program from the get-go. So two-pronged, we use local stocks to minimize the detrimental effect of maladapted or non-locally adapted stocks spawning in with locally adapted stocks. And we do everything we can to ensure that those fish are harvested or got out of the water so that they don't go somewhere else. In fact, we've gone to, since my time in the department, starting in about 2010, is when we started adding the provision into the permits that required them to harvest any fish left in the water after the common property fisheries had ceased.
How do they do that?
The hatchery operators are responsible to hire vessels to go mop up.
Okay. Any questions generated on this slide? Yes, Mrs. Ray.
Yeah, this is one generated by this conversation here. So Sam, if I was to tell you that somebody mentioned to me that they thought only 15% of the hatchery fish that were returning were harvested on average maybe just in general, you would say to me what?
I would, through the chair, I would say show me the numbers, because that's not what our numbers show us.
And I thank you for that.
May I?
Mr. Wood.
To still on slide 5. Without adversely affecting natural stocks of the fish, and your response sounded like that it's not affecting escapement numbers of wild stocks in the areas where the hatcheries are, is how I heard that. But I think that is kind of at the heart of this whole hatchery issue, is are the hatchery fish having an effect on wild stocks, but maybe not in that exact place where the hatchery is, but once, are there ripple effects further out there in other rivers where wild fish would go after they're mingling around out in the ocean and coming back in? So if you're not meeting escapement goals throughout the rest of the state, could that also be attributed to the hatchery fish production. Again, that whole, it seems like, back to Ms. Howard's slideshow, having the information or the science research behind knowing that those hatcheries aren't interfering with the wild stocks returning, it seems to be a priority to make good decisions.
Staff want to respond to those comments? Mr. Raybon?
Yeah, I guess what I would say is that we started locally with evaluating and looking at effects on natural stocks, and we're moving outward. We don't have evidence that shows that there's harm from from hatchery production on natural stocks, especially distant ones. But there is a lot of speculation and a lot of correlation that's driving us to look there and try to get empirical evidence to answer that one way or another. But we don't have data that shows that. We have speculation and correlation, and that's some of the papers that have been talked about around here.
Yeah.
They don't actually have data of their own. They're just looking at other things and looking at correlations and then speculating.
Okay, thank you. But if I may just follow up, I think I'm glad you're listening to that and hearing that, because speculation and correlation could lead to a lot of the answers that we're looking for, and out there. And I'm glad it's not being dismissed. Thank you.
Mrs. Swenson?
Through the chair to Sam. So in some of those hatcheries, are there the terminal streams that for the natural fish, are they far enough away from those so that, when you're harvesting those fish out there, it's pretty hard to tell when the guys are out there with nets whether they're getting hatchery fish or natural fish. So I'm assuming in those areas that the streams, that there aren't hatcheries that close to natural streams. Is that correct?
Through the chair, we have what we call the pre-76 rule. It's in statute AS 1610-400, I believe. And it says you can't put a hatchery on an anadromous system. And so other than the pre-76 hatcheries, which were all fish and game hatcheries that didn't follow the same rules that we have for PNPs, what we're looking for is basically a bay with a waterfall that prevents fish from going into a river. That's a perfect hatchery site. And that's why a place as vast as Alaska has, 24 hatcheries in the entire state? There's more than that on the Columbia River alone, because they don't follow our rules. We have different rules. So a perfect scenario, like I just said, is a system that has plenty of water but no fish in it. And by design, that's a terminal harvest area where you could release fish that come back to that bay and can be pretty much discreetly harvested there, like Lorna was explaining, when we sample the harvest and find very few non-marked fish. It's in the mixed stock fisheries, before they get to a terminal area, where you're going to harvest both naturally produced and hatchery produced fish together. And that's where you have to be really careful. Prince William Sound is a great model of how we do that. We open those mixed stock areas, and every time there's an opening, we're sampling those fish and getting results within 1 or 2 days of what the composition of that is. So we couple that with in-season escapement monitoring on the wild systems and what those proportions are to let our managers know to either fish more aggressively if there's high proportion of hatchery fish and good wild escapement, or fish more conservatively if it's conversely higher proportion of wild fish and maybe not as good of escapement. So we can throttle our fishery management. Commercial fisheries are closed unless opened. It's the converse of sport fisheries generally. So we tell them when they can fish and where they can fish, and we can shut them off really quickly if we find that we're over-harvesting wild stocks when we're trying to go after hatchery stocks.
Okay, thank you.
Any other questions related to slide 5? We move on, I move on, I should say, to slide 6. It's not numbered, but it is number 6, titled An Integrated Approach. Lorna, once again, going back to the results of that Prince William Sound Science Committee, I guess, or whatever their proper name is, that found that 70 to 80% of the catch was hatchery-related in the lineage. And yet you're talking about an integrated approach that would incorporate wild salmon being propagated into the future. Just running percentages mathematically, you're going to run out of wild stock if you haven't already. That's available in sufficient numbers to continue with that mode. So you're going to end up with virtually all hatchery at the end of the process. How is it we're going to maintain a pure wild stock?
I'm not sure there is such a thing as a pure wild stock. If you're talking about an individual stream, because all salmon are straying, so they're mixed wild stocks in the salmon systems. And when you have hatcheries, that's another component of fish that are derived from those same wild stocks.
I'm talking in context of wild versus hatchery.
Okay, so in the case of Prince William, I don't know where your numbers are coming from or what numbers you're referring to, but I would point out that in the last, I think, decade, 12 years, we've set 4 new wild escapement records in Prince William Sound pinks. So natural productivity doesn't seem to be decreasing. In fact, it's setting records. So that tells me that our management strategy of targeting wild stocks through marking, and if it weren't for otolith marking, we wouldn't be able to do this.
Yeah, the study was from the Prince William Sound Science Center, and it dealt with chumps and pinks, not kings.
Prince William Sound, are you talking about the Hatchery Wild Interaction?
I'm sorry, say again?
Alaska Hatchery Research Project?
I don't know what the name of the project is. It was their analysis of the 2013 through 2016 catches that they did each year.
I'm going to have to defer to Bill Teplin on that because I'm not aware of that unless that's our ocean entrance sampling project. I don't remember the years anymore. It's all blurring.
Mr. Chair, for the record, my name is Bill Templin. Can you repeat the numbers that?
They did an analysis of the 2013 to 2016 test catch that they did each and every year, and the results were that 70 to 80% of the chum and pink catch showed presence of hatchery fish in their lineages.
I'll have to check that report that you're referring to and get back to you. I apologize, there's some mixtures in there. The HRP, the Alaska Hatchery Research Program, did a test fishery at the mouth of, at the opening of Prince William Sound for 2013, 14, and 15. That's what I originally thought you had talked about, in which it found large proportions of the return to Prince William Sound were hatchery fish.
That might be it, yeah.
And if that's what you're referring to, that was actually not unexpected, because when we get to the harvests, then we see that most of the fish that are caught happen to be hatchery fish. So we expected there to be a large proportion of fish. And that was kind of the intent. This is the run. These are the adults coming back into the system. By measuring that, then we can look at the numbers of, the proportions of fish in the streams and the catches at the hatcheries and in the commercial fishery, and split all of those by using otolith marks into hatchery and wild. And that allowed us then to create a complete run reconstruction of pink salmon and chum salmon in Prince William Sound for 2013, 14, and 15. It was the first time that we'd actually been able to calculate numbers like the total number of hatchery fish returning to the Sound, the total number of wild fish, the true escapement of wild.
Okay, thank you. Any other questions from board members concerning slide 6?
While we're on slide 6, I just would like to note at the bottom of the page that today there are 2 state-operated hatcheries, 30 PNPs, and 3 federal. And I did a tour of the Jack Hernandez one a few years ago with then Commissioner Cotton and got the tour and the cost of the gas and electricity and the amount of money that it takes to operate these hatcheries. It's a massive business. And just recognizing that there are 30 of the PNPs here, I just wanted to bring that, since we're on that slide, to the attention.
Yeah. And then you and I, Mr. Carpenter, looked at Kodiak Hatchery this year. Which leads to slide number 7. I just want to confirm something. The private nonprofit, do they raise anything other than chum and pink? Mr. Rayburn?
Lorna could answer this, but I'll answer it. All 5 species.
It's all what?
All 5 species.
Oh, all 5. Okay. Learned something. And the state, the 2 state ones, Mr. Payton?
Yeah, primarily Chinook and coho, and then resident species, trout, dollies, things like that.
No pinks, no chums.
Not to my knowledge, no.
Okay. Just wanted to confirm that. And on slide 8, at last year's meeting that we dealt with the herring issue, there was some local and traditional knowledge given to us that operation of 2 of the hatcheries down here indeed did have interaction between the hatchery fish and natural fish in the form of herring, in that they were releasing herring, not releasing herring, releasing the salmon from the, boy, I'm going to butcher this, Medvedchik, how do you pronounce it, down at Deep Inlet?
Medvedchik.
Close enough.
Yep.
That were, when they were being released, timed with the larvae of the herring, were eating up a lot of the larvae. And then also with Neats Bay Hatchery, the dogs coming back were eating the herring likewise because of the timing in the intersection. Is any of that looked at when you making your decisions as to whether the hatcheries are going to be located in a specific locale or whether they can continue an operation at the same levels? Either Lorna or whomever, yeah.
Thank you, Mr. Chair. We're primarily looking at other salmon stocks. I would point out that the southeast herring stocks are, I think, as high as they've ever been recorded in the last couple of years. So we don't seem to be cropping them down too much with hatchery production. The biomass of herring in southeast Alaska is more than we've ever seen.
If you remove Sitka Sound, would that still be true?
There are a lot of local stocks that have either moved or been reduced and not come back. They seem to be transient. But Sitka Sound is the main driver in southeast Alaska.
Okay. Any other questions related to slide 8? Mrs. Ray?
Slide 8, I don't know, I hope this is related to it, but it brought up this thing in my mind here. This would be for Sam. If wild stock, if returning hatchery fish stray into, a wild stream with the wild stocks, how many, and they spawn, and produce some eggs and stuff, and I guess they'd have to, spawn with the wild stocks, the hatchery fish and the wild stocks for this to happen. But like, how many generation, are they always distinct genetically? Like if say after 5 years of, wild fish and hatchery fish, mingling and spawning and, all that happening, would you be able to go to that stream and tell that, a fish is, it was at one time somewhat, or was it sometime a hatchery fish, you know what I mean? Can they just, how well can they sort out the genetics on that after a while? I mean, I remember something I heard that, after a certain number of generations, you can't tell the difference. You don't know, like if a stream, you can tell like a hatchery fish that is found in a wild stream, you can tell that that's a hatchery fish, but only so long. After a while, it's just, those genes are just mixed right in with the wild stocks. Yeah, just talk to that.
Through the chair, I may have to phone a friend here, but that's a multi-pronged question and answer. So first of all, we purposefully used local stocks in our hatchery stock development so that they are from the nearby systems. So they are genetically the same. One thing that we guarded against by doing that is what we call domestication. But because, for example, our pink and chum are reared for such a short period of time in fresh water in the hatchery, the domestication effects are pretty minimal on those. They would be greater with the Chinook and coho, which are reared for a whole year in freshwater. That said, when they do spawn into, when they do stray, and they do stray, all salmon stray, so there's no disputing that. When they do spawn with their naturally produced cohorts in a stream, for example, they're of the same or very similar genotype. They're not genetically distinguishable. And the other thing that happens is selective pressures. So every stream has its own environmental parameters, temperature, flow, things like that. And if a stray, whether it's from a hatchery or another system or whatever, isn't adapted to that environment, they're generally not going to do as well. So they will produce fewer, if any, offspring. And so that potential deleterious effect could be removed just because they're not successful, so to speak. What the Alaska Hatchery Research Project has shown us in Prince William Sound is after it's been almost 50 years now of releases in Prince William Sound of pinks. Program started in '74. There is still stock structure across the sound, and pink salmon have a very shallow genetic stock structure. They're very closely related across the entirety of the range. I'm not a geneticist. My experts over here can talk to that at length. But suffice it to say that they're very similar whether they spawn in Washington, Prince William Sound, or Russia. They're not hugely different. Now, if they were, say, a sockeye that goes up a river and spawns in a lake, then they're very, they're not homogeneous. They're very distinguishable. You can tell those apart really easily. If you had a fish that, say, a chum salmon that spawns in Canada, fall chum, they're easily identifiable and different from a summer chum that spawns in the lower portion of the Yukon versus other summer chum in Kuskokwim, Bristol Bay, Norton Sound. Those are all almost indistinguishable. Well, technically they are indistinguishable. So it depends. But if we're talking about pinks and chums, they're from local stocks. We've demonstrated through our studies that stock structure remains, so they're still distinguishable. And you generally can't tell apart genetically a wild fish from a hatchery-produced fish in Alaska because we use local stocks. I hope that answered that.
Through the chair, thank you very much for that.
Mr. Godfrey?
Sam, I want to pull that thread a little further if we could. I've generally been on the assumption that the straying of a hatchery fish into wild stock and following that into the breeding stream is largely inconsequential. But with what you just said there, they tend to be less hardy if that's not where they originated from. So they may not produce any progeny or may produce less, which in a way kind of seems like genetic selection at work, where man intervened and did something unnatural, creating a hatchery, that nature says, well, this isn't where you came from, so you're not going to fare well. But the only other thing I'm wondering is when they do produce offspring and their progeny go forth, is their progeny as hardy as their peer group in that river, or does it carry on any of the deficiencies of the hardiness of its sire?
Through the chair, I could go on this. We have three more presentations here that will deal pretty directly with those kind of questions. If you want me to stop talking, and we can let others. Okay.
Yeah, just to reiterate what Ms. Carlson Van Dart suggested when she presented, if there's something coming up in a future presentation we'll cover it, just say that, and we'll defer to that point in time. Okay. Any other questions on that slide? I just have a couple on process very quickly. On slide 17 and actually 17, 18, and 19, the board has a very limited authority when it comes to dealing with hatcheries, but one of which is related to egg take. Who determines what the egg capacity of a particular hatchery is?
And a P&P corporation will apply for an egg capacity that's reviewed by in this extensive process, and the hatchery P&P egg capacity is then approved by the commissioner, depending on department review, RVT review.
Does that ever become something that is considered by the board?
No.
I'm sorry, did you say no?
It has not.
Interesting. Okay. On slide 17, you go through the process, and I understand the department takes care of those bullets that are underneath the department segment, of which one is the assessment of production potentials. And then you go to the next step, which is a review process by the regional planning team. Is the regional planning team authorized to initiate items? For instance, let me give you an example. Let's assume the department wants to set production potential at a certain level, and yet the review team wants to go otherwise. Does the review team have the ability to do that, or are they confined to just what the department sent to them?
Right, regional planning team is advisory to the commissioner. And so the regional planning team recommends, for Southeast, for example, production among hatcheries. The P&P permits and fish transport permits and the permits that a hatchery needs to operate are approved by the commissioner. And so regional planning team recommendations are taken under advisement by the commissioner.
Okay.
A lot of this process, yep, go ahead.
So being advisory only, they have no ability to make any modifications. So then we go to a public hearing, which would be the next step, correct?
Mm-hmm. Yes.
And then after the public hearing, the commissioner does what he's going to do or she's going to do, and no one else has any ability to change that direction. Is that correct?
Right, yep. So
I'm trying to figure out where does a board's role with egg come into the process?
Sam, would you like to speak to that?
Mr. Raybone.
Thank you, Mr. Chair. AS 1610-440B has a provision that after a permit's issued, the board can, a plain reading of it says the board can adjust the source and number of salmon eggs.
Yeah, and I think legal has interpreted that to mean both hatchery eggs and wild eggs, if you want to call them that. All eggs are covered under that proviso. But where, if it's not in the application process, in what process would that take place?
It hasn't happened yet, but I assume it would be a board proposal and then it would be likely addressed at some point. But it hasn't happened to date because background of 1610-440B, that was written before there were any hatchery brood stocks. And all the egg takes were from wild stocks. And so it was allocative to take fish out of a system. And so that's why it was given to the board to amend a hatchery permit to the source and number of eggs.
I understand that's a Mason and Ashburn argument.
Yes.
Yeah, okay. I think that might conclude all of mine, and everybody will sigh relief. Oh, just one last one is also with process. Slide 25, the permit alteration request. I never heard that term before. Who can initiate that? Who's authorized to initiate? Can a private citizen initiate something of that nature? Lorna?
So, to date, all permit alteration requests have been submitted by the P&P Corporation Board of Directors. Let me look at 5A, C 4850 really fast to confirm that only the
Yeah,
P&P
and
Corporation.
if you do so in the context of my question, I'm trying to find out who's that process available to for use.
It's the P&P permit holder who does the request to alter their own permit.
Nobody else, correct, Mr. Raybone?
Correct.
That's what I'm trying to find out. That concludes all my questions, and I thank the indulgence of the other board members. But this is a process that's been going on ever since I've been on the board, and I'm trying to get to understand it fully. Any other questions, Mr. Wood?
Chair, if I may, I just would like to point out slide 22, just in the salmon enhancement tax last year was $8,500,000, and then $6 million the year before and the year before that. So really large amounts of money focused on these hatcheries. And I guess I'll have more questions about that down the road as we go throughout the day, but that really, after my tour of the hatchery and then looking at these numbers, it always strikes me.
Any other questions? Not, let's move on to the next presentation, which I understand to be the Sports Fish Enhancement Program, and that'll be Chuck Pratt. Anytime you're ready, go ahead and proceed. This is something you need to get involved with, a technical one? Okay. I would probably make it worse. Okay.
You ready? Oh, yeah. I was just making sure. Good morning, Mr. Chair, members of the board. It's a pleasure and honor to be here today. My name is Chuck Pratt, and I'm the Hatchery Program Supervisor for the Division of Sport Fish. Today, I will be discussing the Sport Fish Enhancement Program, its mission, and relationship to Alaska's private nonprofit hatcheries. Sport fish stocking in Alaska began in the 1950s with the release of rainbow trout into lakes along the road system in south central and interior Alaska. Sport fish stocking is intended to create and diversify sport fish opportunities in support of the division's mission to protect and improve the state's sport fishery resources. Today, the Division of Sport Fish operates 2 state-of-the-art hatcheries, the William Jack Hernandez Sport Fish Hatchery in Anchorage and the Ruth Burnett Sport Fish Hatchery in Fairbanks. ADF&G also owns and pays for much of the operation of Crystal Lake Hatchery near Petersburg, operated by the Southern Southeast Regional Aquaculture Association, and funds sport fish enhancement activities at the Kodiak Regional Aquaculture Association's Pillar Creek Hatchery on Kodiak Island. The sport fish stocking program is responsible for the release of 7.2 million fish each year, 2.3 million of which are produced by the Pillar Creek and Crystal Lake hatcheries under cooperative agreements. The enhancement program is comprised of 2 distinct elements: smolt stocking and lake stocking. Species produced may include rainbow trout, Arctic char, Arctic grayling, lake trout, king and coho salmon. These may be as fry, fingerling, smolt, subcatchable, catchable, or broodstock. Production of Arctic grayling was suspended in 2020 due to budgetary constraints. Currently, the sport fish hatcheries are at or near maximum capacity for most species. Changes to statewide stocking plan usually require reallocation of existing production from one site to another, as there is little to no opportunity to increase production. Recent changes in the production of lake trout at the Ruth Burnett Hatchery every other year, which began in 2022, the temporary suspension of Arctic grayling, and the discontinuation of the Deception Creek King Salmon Enhancement Project on the Susitna system. An increase in catchable kings for ice fishing on the Chena Lake in Fairbanks is projected to begin in 2025. Regulation of sport fish hatcheries is consistent with private nonprofit regulation, with the exception that a hatchery permit and basic hatchery management plan are not required to establish a sport fish hatchery. Sport fish hatcheries are required to submit annual management plans, annual reports, and secure fish transport permits for all releases, and follow state genetics and fish health policies. One distinctive element is the statewide stocking plan. This plan outlines the division's stocking objectives for the next 5 years and is updated annually after public review and comment. It also contains the division's lake stocking policy, which describes the conditions under which a given location may be stocked. This plan effectively replaces the hatchery permit and basic management plans required for private nonprofit facilities.
The Sport Fish Enhancement Program is unique in providing statewide fishing opportunities for numerous species in a variety of water bodies. Fish produced at sport fish hatcheries are released at over 240 sites across south central and interior Alaska. The sport fish funded private nonprofit hatcheries in Kodiak and Southeast contribute to over 20 more. Many of the king and coho fisheries are designed to have 100% of returning anglers or of returning adults harvested by sport fish anglers. Private nonprofit hatcheries support sport fish enhancement through cooperative agreements to produce and stock in areas not easily served by sport fish hatcheries in Anchorage and Fairbanks. This includes stocking in Kodiak, Juneau, Petersburg, and Ketchikan. Other private nonprofit hatcheries across the state support sport anglers through a variety of stocking projects not funded by sport fish enhancement. Fish returning to those release sites are available to anglers through common property fisheries. Total statewide production of king smolt in 2022 was approximately 10 million from all hatcheries. The Division of Sport Fish Hatcheries and the sport fish funded private nonprofit hatcheries accounted for 40% of the smolt released. There were 28 sites across the state where king smolt's releases occurred in 2022. 15 of the sites were funded through the Sport Fish Enhancement Program and represented the only king smolt production in Cook Inlet, Resurrection Bay, Prince William Sound, and Kodiak. In total, there are 24 sport fish funded release sites where king and coho are released. Some sites, such as the Seward Lagoon and Menasha Creek, receive both king and coho. While the Division of Sportfish funds releases of king and coho salmon at Pillar Creek and Crystal Lake hatcheries operated by the Kodiak and Southern Southeast Regional Aquaculture Associations, the William Jack Hernandez Sportfish Hatchery in Anchorage is the only sportfish hatchery producing king and coho salmon smolt. Each year, the Hernandez Hatchery releases approximately 3 million king and coho at 11 sites across South Central Alaska. The sportfish hatcheries, Hernandez and Burnett, produce 4.8 million fish, or 330,000 pounds annually. In Alaskan units, that's 316 cow moose. While the 62% of the fish produced are for anadromous release, 70%, or 230,000 pounds, that's 164 bull moose, are destined for the freshwater lake stocking program. The lake stocking program releases approximately 1.8 million rainbow trout, Arctic char, Arctic grayling, lake trout, king and coho salmon into more than 240 sites across Alaska. Most of these fish are sterile, so they are unable to reproduce in the wild. This is done to minimize the potential for illegal transport and possible establishment of feral populations that could damage the wild stocks. While the Sportfish program has limited research capacity, it does work to evaluate and characterize the success of its projects. 100% of the kings and coho smolt released are otolith marked. Additionally, king smolt released alongside wild populations are adipose clipped, adipose fin clipped, to allow visual identification and segregation of returning adults. All broodstock used to produce future returns are tissue sampled each year to aid in future genetic analysis. The king smolt released at Ship Creek are being evaluated in a 10-year study aiming to improve smolt to adult survival and more closely reflect the age structure of wild populations. Furthermore, the spawning of king adults by size is being done to reflect the current findings in male to female size relationships of wild matings. As sport fish enhancement is intended to create and diversify angling and harvest opportunities while minimizing impacts on wild stocks, fish are primarily stocked with few or no native fish present, so that 100% of the fish may be harvested. This strategy is evident in southeast Alaska on inside waters, where king enhancement activities are distanced from wild stocks as much as possible, so that management restrictions applied to protect wild stocks do not limit angler access to enhanced adult returns. Both Crooked Creek and Ninilchik River are managed as integrated enhancement projects with the objective of limiting genetic impacts on wild populations by only releasing smolt produced from wild parents at these sites. Hatchery produced smolt released at Crooked Creek and Ninilchik River are 100% adipose fin clipped, allowing managers to target returning hatchery fish in sport fish hatchery or in sport fishery, while limiting harvest of wild fish to achieve spawning escapement goals, a strategy often referred to as a mark select fishery. During the current time of low king salmon abundance, the structure of the king salmon enhancement program has provided the opportunity for fishing and harvesting kings not otherwise possible due to fishing closures on wild stocks, and particularly in south central Alaska. Fish stocked in lakes are primarily sterile to minimize genetic impacts in fresh water. Projects are evaluated to ensure that they are fiscally responsible based on sound scientific practices and provide benefit. The funding for sport fish hatchery operations is made up of a combination of Alaska legislature appropriated general funds, proceeds from sport fish license sales, including king stamps, and federal aid called sport fish restoration funds, which are derived from taxes on fishing equipment and motorboat fuel. The fiscal year 24 projected costs for the sport fish enhancement program is $7.2 million. Northern Economics conducted a study in 2004 to assess the benefit of sport fish program in Alaska. Angler effort accounted for nearly 330,000 angling days and $34 million in spending each year. Adjusted for inflation to 2023, the economic value of the sports fish program becomes $55 million. In summary, the enhancement, sport fish enhancement in Alaska predates statehood. The Sport Fish Enhancement Program seeks to create and diversify fishing opportunities for anglers in both fresh and saltwater while minimizing impacts on wild stocks. The program includes resident species not produced at private nonprofit facilities. It is subject to much of the same oversight applied to private nonprofit aquaculture associations, and is funded by a blend of general funds, fishing license sales, and federal taxes on fishing equipment and boat fuel. But lastly, The Alaska Department of Fish and Game's Division of Sportfish Enhancement Program provides an economic, recreational, and consumptive benefit to anglers. Before I'd open it up to questions, I'd like to offer all members of the board to meet with me at any time down at the William Jack Hernandez Sportfish Hatchery for a tour and further discuss sportfish hatchery operations. So, are there any questions?
Thank you. Any questions from the board? Mr. Carpenter?
Thank you, Mr. Chair. Thank you, Mr. Pratt. First of all, I'd like to state I'm a really big proponent of the state's enhancement programs. It provides a lot of opportunity in many, many locations around the state, and so thank you for your good work down there. My question is specifically addressing king salmon that you raise. And I know they're very expensive to raise, and it takes a long time to get a return back, and it's a very important species for anglers, not only recreationally, but for subsistence reasons in certain areas. Is the state currently maxed out on the amount of king salmon they could raise due to incubator space, raceway space, things like that? Or is it a function of just not having enough money in the budget to increase that production?
Oh, why don't we just stop now? Gonna have to stand down. We have a technical problem. Apparently, the recording has stopped. Want to do a lunch break now? What time is it? Oh, good timing. We'll just go on a lunch break here and make Matt Miller come back and visit with Mr. Pratt and us.
And do I get to repeat my presentation again?
I think we can dispense with that. So why don't we say 1:15, return to meeting? Can we do what? I can't hear you. 90 minutes. 90 minutes, okay. Well, tell me a time you want.
1:30.
What? 1:30? 1:30 it is, folks. Ladies and gentlemen, we will be ready to go. Where we left off was, are there any board questions of staff related to the last presentation of the Sports Fish Enhancement Program by Mr. Pratt? Any questions from the board? Mr. Carpenter.
Thank you, Mr. Chair. I'll make it real quick. I asked my question, and you don't have to elaborate dramatically because we are a little bit short on time, but could you just give me the king salmon hatchery production and the sport fish hatcheries? Are you at 100% capacity in regards to rearing fish?
Through the chair. No.
And just a quick follow-up, is that because of it's harder to acquire brood nowadays, or is it a factor of cost?
Through space. We have a small increment that could be implemented. So we're not at ultimate maximum capacity, but as far as operational, we're at maximum capacity.
Any other questions? Like Mr. Carpenter, I don't have so much of a question, but a comment. I live, when Deception Creek is up and running, I can actually hear from the house. Mr. Miller came out when that decision was made to discontinue the weir. The fish are not there, but that could just as easily be attributed to fish on anywhere else either. But you've issued an invitation for us to come to you. I'm going to issue an invitation to you to come on out next summer and take a look for yourself as to what is or isn't occurring. And one of the unintended consequences of closing the weir, which I was in favor of, by the way, is that we no longer have brown bears coming into the yard quite as frequently. In fact, we haven't seen one in about two or 3 years now, and before they were constantly in and out. So, you know, when you stop fish, there's this consequence in nature, and we're seeing it lived out. And I just wanted the department to be aware of that. So, and I would like to see you out there next summer, maybe even Matt if he's still around. With that, we'll go to the next presentation. I believe Mr. Templin's up next. Hatchery Research Program Overview Results. Correct, Bill? That's the order I have, man.
You ready? Okay. Good afternoon, Mr. Chair, members of the board, and members of the public who are here. Thank you for staying for so long. My name is Bill Templin. I am the Chief Fishery Scientist for salmon at the Commercial Fisheries Division of the Alaska Department of Fish and Game. With me here is Sarah Gilk-Balmer, the department's new principal geneticist, and we will provide you with a summary review and update on the Alaska Hatchery Research Project, also known as the Hatchery Wild Interaction Study. This is a long-term, large-scale, multi-species, multi-year study that encompasses 2 regions of the state, and which has only been successful due to the contributions of many people and organizations. Now, a decade after it began, results for some parts have been completed and already made publicly available, while other parts continue. This year is the last year of field work. It occurred in southeast Alaska with chum salmon. This will be a relatively brief overview of the project. The information that we will present today has been presented in previous Hatchery Committee meetings, all-day public information meetings, and in reports and publications. Throughout the presentation, we will provide links to those resources to help you find them. The rationale for the expensive time and resources on this project can be found in the Alaska State Constitution. Under Article 8, it says, "Fish, forests, wildlife, grasslands, and all other replenishable resources belonging to the state shall be utilized, developed, and maintained on the sustained yield principle, subject to the preferences among beneficial uses." As the state's entity responsible for management of Alaska's fish, wildlife, and aquatic plants, the Department of Fish and Game's mission statement builds upon these principles to protect, maintain, and improve the fish, game, and aquatic plant resources of the state and manage their use and development in the best interest of the economy and the well-being of the people of the state, consistent with the sustained yield principle. Both the Constitution and the mission statement carry with them the assumption of precaution when decisions are made affecting Alaska's natural resources. The Salmon Enhancement Program was developed with that as its foundational guidance. Many people don't realize that the hatchery program in Alaska is about fisheries. You've heard a lot about that just this morning, so I'll not spend a lot of time on this. It's about fisheries rather than fish. It's designed to provide supplemental harvest and to provide an economic benefit to support Alaskans and Alaska communities and economies. Since the natural production is the first priority, statutes, regulations, and policies have been developed to guide and put conditions on enhanced production to ensure that there is no significant adverse impact on natural stocks. We've gone over the contents of this slide, so I'm going to go right through it, but basically I want to remind you that that hatcheries have been part of the increase in production in Alaska since statehood, and as you can see from this plot, it has been additive to the underlying natural production. The structures built into the hatchery program were founded on the commitment to an approach to enhancement that would provide long-term benefit to Alaskans while minimizing the risks to natural production by wild populations. These principles were described in an understandable way in two previously published papers. First in 2004 by Steve McGee, which provides an overview of the precautionary approach built into the Alaska Hatchery Program. The second is similar, but provides more detail by focusing on Southeast Alaska and that portion of the hatchery program. Both have been made available on the webpage for this meeting. More recently, the policies, procedures, and permitting within the hatchery program and the way that they have been implemented was reviewed and documented in a department report in 2018. Now, this review was also described in more detail in a presentation at the October 2018 Board of Fish work session, and I have the link here on this slide for you to make it easy to find. This report began with an overview of the structure of Alaska's precautionary approach to the development of the hatchery program through forward-looking policies, plans, and permitting. Much of this portion of the report was previously presented in that paper by McGee. The report then lays out the elements of the approach as they relate to the three categories of fishery management. Categories of fish management, fish health, and disease, and genetics. This was described to you this morning in Lorna Wilson's presentation. Following that, though, we provide two case studies that have been presented to provide examples of how this structure works out in actual practice. It's important to remember that these case studies are necessarily abbreviated and are not intended to provide an in-depth or extended review of the program, but they do provide an ability to see how the structures panel of experts to design and implement a long-term research project to inform resource management decisions. The Alaska Hatchery Research Science Panel was formed in 2011 to design a study that would provide the information all stakeholders need to understand potential impacts of hatchery-wild interactions in Alaska. The program was funded through a tripartite partnership, including the state, processors, and PNP operators. The effort focused on two primary species where the least research on interaction effects was available: pink salmon and chum salmon, and the two regions with the largest hatchery production, Prince William Sound and Southeast Alaska. More information is available in this document here. The science panel for this research program designed and has overseen the implementation of the project. Panel members have changed over time, but have all been experts from the department, NOAA Fisheries, Hatcheries, and the University of Alaska Fairbanks. The names and affiliations of individuals on the science panel are listed here, and more details are available on the website. The structure of AHRP is broadly overseen by the science panel. The laboratory logistics and analysis portions of the research program are generally handled by the department, the blue boxes. The Prince William Sound Science Center and Sitka Sound Science Center are both contractors on the project, the green boxes. They have been responsible for most of the field work and collection of data and samples. Contractors have also provided annual reports and authored or co-authored peer-reviewed publications of field-related findings. And here's a link where you can find more of that information. Hatchery origin fish were observed in wild streams, and hatchery applied marks were available to make positive identification. And then again, in about 15 years ago, larger surveys documented consistent presence of strays using those otolith marks. They were found in streams in Prince William Sound and Southeast Alaska. These results raised several important questions, including, are these hatchery origin fish interbreeding with wild salmon? And if so, what is that having an effect on the productivity of these stocks? These and similar questions were considered by the science panel, and the concepts behind the concerns were refined into 3 priority questions that could reasonably be addressed through a scientific study. These questions do not encompass all the needed information, but they were considered fundamental to understanding and also essential. On top of that, they were designed in a way that they were answerable by studies. These questions are: what is the genetic stock structure of pink and chum salmon in Prince William Sound? What is the extent and annual variability of straying? What is the impact on fitness of natural pink salmon and chum salmon? And in this case, fitness is measured as productivity, return per spawner. So when you're comparing them, and Ms. Gilk-Baumer will describe it to you, relative return per spawner between hatchery and wild fish. We had to determine that prior to doing the study so that the study could be designed to get the level of information that we needed. There's more information at this link on the website. Finally, I'll walk through each of the questions and describe to you why they're important. Why did we choose them? The first question was, what is the genetic stock structure of pink and chum salmon in Prince William Sound and Southeast Alaska? Hatchery brood stocks are all derived from stocks within their respective regions, as per the Alaska genetics policy. Brood stocks are necessarily large because of the scale of the program, and broodfish are spawned non-selectively from throughout the duration of the spawning run. As a result, broad-scale genetic variability is maintained, but the distribution of genotypes may differ from wild spawning populations, which are smaller and potentially adapted to local environmental conditions. Policymakers and resource managers need an improved understanding of the hierarchical genetic structure of these populations and the degree of genetic differentiation at and within the level of harvest management decisions. This information can provide insight into changes to stock structure that might be associated with the presence of hatchery production. It also provides the capacity to track future changes in the resource. The second critical question is, what is the extent and annual variability of straying? It has two parts. The first is identifying the patterns and proportions of strays within spawning streams in Prince William Sound and Southeast Alaska. Why is this important? Knowing that hatchery fish are present when spawning happens to be a prerequisite for understanding the risk from interaction while spawning. If the hatchery fish aren't present, the risk is zero. If they're present, then risk exists. Likewise, knowing the magnitude, how many, and the patterns, where and when, of straying into space and time informs assessment of the potential risk. This information can also be used with other information to estimate the number of hatchery origin fish that ended up in the wild spawning streams each year. Part two of this question is about run reconstruction. It involved estimating the proportion of hatchery fish entering Prince William Sound with a test fishery. This is the question that I answered this morning at the mic, concerns about the proportions. Salmon harvest in Prince William Sound is directed mainly at harvest of hatchery origin adults. So, proportions of hatchery fish in the harvest is not an accurate measure of the relative proportion of hatchery fish in the run. So we could not use the harvest information to determine the relative size of the hatchery and wild run into the sound. That's why we needed the test fishery. One benefit of that, though, is that combining the information from the total run, the commercial harvest, the estimated escapements, and the proportion of hatchery fish in the escapement, we could reconstruct the runs for 2013, 2014, and 2015. And this was the first time this was possible, and it enabled us to estimate the following, the total size of hatchery and wild runs, an independent estimate of wild escapement. It gave us numbers of hatchery strays. It allowed us to calculate the number, not just proportions. Harvest rates on hatchery and wild runs separately, and the hatchery donor stray rate. How many of the fish are straying out of the hatchery? This is only possible in Prince William Sound. A similar detailed reconstruction was not possible for chum salmon in Southeast Alaska. The final critical question is, what is the impact on fitness or productivity of wild pink and chum stocks? Fitness, or the ability to survive and reproduce, was defined here as productivity, which is the average contribution of an average individual to the next generation. The rationale behind this was that if hatchery fish are less fit in wild streams, then hatchery fish will produce fewer offspring. And if this lower production can be inherited, then wild fish may end up producing fewer offspring due to that interbreeding. That's a simple statement of the risk question that we were trying to answer. Why is it important? Pretty much because Alaska wild stocks of salmon have priority. You've been introduced to these two sources of wild stock priority. The first is in the Policy for Management of Sustainable Salmon Fisheries, which protects wild salmon stocks from adverse impacts from artificial propagation. And second, in the genetics policy, which talks about protection of wild stocks from possible harmful interactions with introduced stocks. Those harmful and adverse genetic interactions include potential loss of diversity among populations and introduction of poorly adapted traits into the wild populations. Finally, these results have been published and are continued to be published in peer-reviewed journals, with more publications on the way. As we complete the remaining fitness work and finalize the population structure, those two will be published. Why do we publish in peer-reviewed journals? That's because it puts it out to a wider group of experts, our peers in the scientific community, and allows it to be criticized or supported, used, and provides an independent kind of review for the work that's being done. In addition, the website is there. Sometimes it's kind of hard to find. So I've put the link on here for the Hatchery Research website, along with places on that website where you can find reports and publications, as well as science panel, informational meetings, and workshops, the agendas, as well as most of the presentations. Mr. Chair, that concludes this portion of our conversation. If you'd like to stop for questions, fine, but maybe you'd want to hear some of the results before. Sarah might be able to answer some of the questions.
Why don't we open it to questions and we'll carry it forward from there. Carlson Van Dort.
Hi, Bill. Thanks for the presentation. I'm looking at slide 7 and really just kind of noticing things here. And it looks like there's been a pretty steady trend in increasing since about 1990 or so with the non-chum and pink species production being, I don't know, pretty consistent, I'd say. But there does seem to be kind of a, an increase, particularly among chum, it looks like, but also among pink salmon, approaching 2 billion total, it looks like, in 2022. How or does the department assess potential impacts on other salmon species? So, like for pink and chum, the peak of the run, or the coho are coming in after the peak of the pink and chum runs, and I'm just wondering, do you monitor that? Are they using some of the same streams? Are there impacts if these streams are loaded with pink salmon, for example, and coho are trying to get in there after them? Is that presenting any problems that you're aware of? Does the department monitor for that at all?
Through the chair. There are activities on local level, observations by managers and research biologists. But as far as I know, those observations aren't collected. And I'm going to ask the director if he knows.
Well, I kind of spoke to that a little bit earlier today. We're focusing on inner circles and moving outward. So some of Dr. Howard's work is where we're getting into other interactions.
Yeah, I mean specifically as they're approaching the freshwater environment, towards the ends of their life cycles and not necessarily so much in the marine. That's what I was kind of curious about, whether or not you're looking out for that or if that's something that's being paid attention to.
As you might recall, most of these releases are in terminal harvest areas, and a lot of them are barrier systems and things like that. So what we've done is looked at stray proportions and things like that. I'm not sure I'm answering your question adequately, but I mean, those are all on the list of things to do as we check things off,
Yeah, I mean, what kind of brought me to that question is I'm looking at, 2 billion-ish is a pretty substantial amount, I think, and then you couple that with varying stray rates throughout these different regions and whatnot, and I'm just curious whether or not there are impacts on other fish like coho that kind of use some of the same habitat, after the fact, and, in some places coho are having a hard time, so I just was curious about that.
Yeah, just to expand on that, I'm glad you mentioned coho. That helped trigger me. Separation is a key part of our program, and so temporal separation or timing is a big piece of it. So coho being such a late portion of the run, that is a component. We have evaluated in hatchery release location or terminal harvest area requests what other stocks are there. are they index streams and things like that? And we've denied quite a few over the years that we felt like would be a conflict. But, I mean, it's kind of a case-by-case basis, and it's typically during the permitting phase that we look hard at that stuff. And then we evaluate it for the first cycle or two to make sure we're comfortable. And in many cases, we make adjustments when we're not comfortable.
Last follow-up, Mr. Chair. Thanks. Yeah, and my questions are sort of rooted in concern that has been brought to me over these somewhat large volumes of pinks that are returning. They're in streams, they're dying, they're consuming, the decay is consuming oxygen, and then the cohos move in, as you mentioned, the temporal sort of spacing. That was just a concern that was brought to my attention, and I just thought I would ask some questions around that. Thank you.
Any other questions? Mr. Carpenter?
Thank you, Mr. Chair. Thanks, Bill. And if the question is going to be answered in the next slide, just say that in the next presentation. It's specifically to fitness. You know how you use Prince William Sound as an example, because that's what I'm most familiar with. But you said in your presentation that, you know, during this study, you're looking at if the hatchery fish that were straying in close proximity to the hatcheries were being out-competed in the streams by wild fish, and that the possibility of those traits could possibly be adapted by wild fish in the long run. Has the wild stock returns for pink salmon, using Prince William Sound, for example, been diminished, let's say, over the last 10 years, even with the findings, you know, in regards to the straying percentages around the hatcheries?
Through the chair. No, they haven't, but that's a qualified no. The odd year run is doing much better than the even year run. When we have these discussions, there's a distinction that needs to be made, and sometimes they're equated. One is the total return, the number of fish that come back, and often that's called the productivity, but then there's also the productivity measured at how many fish came back per spawner, right? So that's how many fish were produced by one spawner in the past on average. So those two numbers sometimes get conflated. So yes, we've had big runs, but we've also had good escapements. So one interesting thing is that we had the die-off in 2019 with the drought and a lot of fish dying in the streams, and then in 2021 we still had a really good pink salmon run. So it's highly variable, and I'm not sure how you would be able to detect unless we're looking at fish per spawner, that productivity, which is the measure we used here. It's difficult to distinguish whether there's been an effect, or even if there's a difference between hatchery and wild fish spawning in the wild. And again, that's why we have this very expensive, long-running experiment or study. I don't know if that answers your question completely.
Oh, thanks for that. Yeah, it answers my question. I mean, I'm just, you know, I look at some of these hatchery programs, and, you know, for example, the Prince William Sound hatcheries have been there for about 50 years. Pink salmon return in 2-year cycles, and so you have many, many, many generations of fish that have been evolving with wild stocks in Prince William Sound over the last 50 years. And, you know, if the traits of hatchery strays in Prince William Sound were to have done detrimental harm to the wild stocks in the streams in Prince William Sounds, which produce less offspring, you would think over that long of a period of time you would be seeing really reduced wild stock returns. And I personally haven't seen that, and that's why I asked the question.
Are you through? Yeah. Any other questions, Mr. Godfrey?
So I want to revisit the topic that I brought up earlier that once you addressed here, when hatchery fish are generating offspring with wild stock in the rivers that they were not originally reared from themselves. Obviously I'm not a geneticist. I probably couldn't even spell geneticist, but Is there any theory, are you aware of any theory or evidence that there could be any benefits imparted to wildly reared salmon in stream from one of its parents being hatchery fish?
Through the chair. That's a really good question and an often asked question, because often people consider it not fair to only consider the risk, the harm, and not consider the potential benefits. Unfortunately, the most probable effect is going to be in the risk side, if there is an effect, because we're talking about fish that were the only difference between them and their brothers and sisters, or their distant cousins in the wild streams, are the fact that their parents may have come from the hatchery, the grandparents came from the hatchery, their great-grandparents back 25 generations. So over time, they may have accumulated something. We haven't measured it yet, but yes, the theory is basically that if there is an effect, it's going to be negative because the hatchery fish come from this long line in a different environment, while the wild fish have been going back and forth with whatever the selective pressures are in the environment. So I do believe it should be considered, but it's one of the less likely potential results.
Okay, thank you. And then one other question along the similar lines, and you may not know the answer to this. It may not be knowable, for all I know, with current technology and methods, but when a hatchery fish is strayed into wild stock and it's in stream and it's spawning, presumably with the wild salmon, the odds are probably it's not another hatchery fish. In the biology of salmon, is there a tendency, is there evidence or way of knowing certain traits are predominantly or universally imparted from the mother or the male, one or the other in that instance? Well, I'll give you, for the sake of simplicity of what I'm trying to ask here, in humans, it's imparted by the sire. So we know that. I mean, sex, gender, okay? So we know that. But I think there's lots of other things geneticists are just learning is generally imparted by one or the other based on the chromosome contributor. But is there anything along those lines that we're aware of with salmon? The fertilizer versus the egg layer?
Through the chair. Yes, there are differences that are inherited, but we tend to think of them as sex-mediated. In other words, not sex-determined, because every time we think that we've got something determined, then we find that it's not. But definitely, there are traits or expressions of a gene that are related to whether or not it's a male or salmon is a male or a female.
Thank you. But I wasn't talking specific to the gender. I was just talking about certain traits. For example, size. Is size imparted beyond environmental factors? Is the size that's in the DNA of a salmon, is that determined predominantly by the fertilizer or the egg or something like that? But that's fine because it's probably not really that. Maybe we'll know a lot in 100 years on that, but it's probably not really relevant. My only other question then is, when it comes to, say, genetic selection in nature, self-terminating and stopping the propagation of certain genetics that are less desirable from nature's perspective, would in a general theory, would it be reasonable to assume, absent other data, that when a hatchery spawner is in wild stock and spawning, that, to what was said earlier, sometimes they won't even spawn, they're less vigorous, less hardy for the environment, and they probably are going to produce less eggs or fertilize less eggs, if at all. Wouldn't that also tend to mean that, if that was the case, that the progeny is going to carry some genetic expression from that moving forward in one way or another? And I guess what I was getting to when I was asking, is it the egg or the fertilizer that imparts something? One of them I was getting at is the hardiness, the vigor of a little tiny salmon before it goes out in the world and takes on other environmental challenges. Is that something that's dictated, if it's known, by the female or the male? And then that's where it just led me to my other question here. And I know it's getting deep, and it's probably not that. It's just my curiosity.
Mr. Chair, through the chair, Sarah's going to show some of the results of the relative return per spawner that will demonstrate that there are differences between the sexes of hatchery fish and how productive, how many offspring they produce. And we had, all of our answers have to kind of go through that narrow funnel of what we're actually measuring. But there is a difference of whether it's a male or a female that came from the hatchery, especially if they've got mixed heritage, hatchery and wild. So there is a difference, and that difference does seem to be mediated if it's a female hatchery fish spawning with a male natural fish or vice versa. The other part of that then also is sorry, I just blanked on my answer, but maybe it'll come back. But Sarah's going to demonstrate some, and maybe from the graphs that she shows.
Thank you.
That question come back.
Appreciate it.
Any other questions? Yes or no? Okay.
No, I'll wait.
Thank you. I just have 3 quick questions that don't go into detail. And other than lawyers, scientists, and academics seem to have longer answers. I'm going to try and keep it short and concise. The reconstruction data that you were referencing, would I find that, because I'll go into weeds, will I find that in the references you put towards the end of the presentation on slide that doesn't have a number with the public and professional context slide? Is that where I find that information?
Mr. Chair, yes, you would find it there. You would find the reconstruction in.
Would you just send me the link to that?
These papers are available online, but I'll send you the link.
Okay, thank you. The next question is the peer review process. The RC2, which is just a cover sheet of the article from diatoms to killer whales, impacts of pink salmon on North Pacific ecosystems by Dr. Rugarone and 7 others. Has that been peer reviewed, or is it going through the process of being peer reviewed? Do you know?
Mr. Chair, that has just recently come out. It's been through peer review, and it's now published. The paper that you're talking about by Greg Rugeroni et al.? Yes.
And have you read that?
I have read it, but it's going to take a longer read. It's 30 pages of work, and it's going to take a finer read and a more comprehensive read than I would normally give to a paper, just because it's important to this whole discussion that we're having, as well as the wider discussion in the ocean.
I totally agree. I spent probably a good day on it so far, just trying to wade through it. There are some conclusions in there, and I'm not going to go through all of them, but there seemed to be correlation across the board. Everything that he picked up, there was correlation. And you're somewhat famous for a couple of years back. Correlation does not equate to causation. What does it take to jump from all of these correlations to a causation status?
Mr. Chair, again, I've got a friend in the back who's counting how many times I say that's a good question. But that is a very good question. It's very well directed. If you remember Katie's slide, she had a slide where she talked about 4 aspects of a correlation that give it strength, right? And that actually comes from Bradford Hill. It was published, those 4 were part of 9 points to look at in a correlation study by Dr. Bradford Hill for use in healthcare. So it's also like diagnosing an individual. Here we're diagnosing an ecosystem. And that the number of correlations and the number of species that you see that correlation occur in increases the strength of the potential that it's a real factor. However, and here's my quick, try not to give you a long answer, but these are almost all of the papers in that paper, the Ruggiero paper, almost all of the studies that are cited are using a single data set, which is the Ruggiero and Irvine compilation of the number of salmon and the biomass of salmon in the ocean. So they all use the same data set. Now, department realized that that was very important, understanding that data set and confirming it. So the department now is involved in trying to recreate that and extend it on into 2023.
Yeah, and I did ask her when that presentation was made, does this not cover 3 of the 4? And the response was affirmative, that it does touch on 3 of the 4. Somehow I want to get to where we can see a clear need to do research into what impact the pinks are having. And that's where I'm going and trying, at least that's where I'm trying to go. I'm not being real successful at it, but that's where I'm trying to go.
Mr. Chair, I agree with you. The department agrees with you. There's sufficient in here for us to be taking a look. I don't believe, and this is my personal professional opinion, that there is sufficient in there for us to start acting tomorrow. I think that's one of the big reasons why we're spending considerable amount of scientist time trying to recreate the numbers, to figure out where the numbers came from, as well as review the papers, as well as go out to sea and try to sample so we collect data that can be used to compare to the results of these studies.
This is the most encouraging testimony I've heard thus far during this meeting. Thank you. And there's reason for concern. You know we're dealing with Yukon issues, et cetera, and what's happening out there. And one of the results was analysis is quantitative analysis of 47 sockeye salmon population representing 90% of all sockeye. And where he goes with that, he says an estimated increase of 119 million in pinks abundance would be associated with a 9% decline in sockeye productivity in the Bering Sea and the Gulf, and a 21% decline of productivity in BC and Southeast. And it goes on with the mean annual return during the period of 05 through 15, et cetera, was estimated to have reduced sockeye salmon productivity by 5% in the Bering Sea and 9% or 6% in the Gulf. I mean, those are pretty definitive conclusions. And if the people on the Yukon want to look at a possible effect of some of the things happening there, this may very well be a good starting point. And that's where I'm heading, okay? So, yeah, if you wish to respond, you certainly may, yeah.
Mr. Chair, I am familiar with the paper that you're referencing there. I disagree with the results, and that's for some technical reasons, and so I'm not going to get into it there. I think maybe there's a demonstration of something occurring, but I think that extrapolation to get the percentage of decrease in the productivity of sockeye in BC, that's something the biometricians and the scientists that I've asked have not been able to understand that the calculations that went into that. So my colleague, Dr. Andrew Monroe, is working with Brendan Connors and others that are on that paper so that we can begin to understand how it was done and maybe make sure that the numbers that are used are ones that the department could also stand behind.
Yeah, I really appreciate that. One thing should be crystal clear, I am not a biometrician, okay? I'm trying to read all this, digest it, and use common sense to come to conclusions. With that, I'll go forward. Any other questions?
If I may, I'll be quick. I just would like to put an exclamation point on Chairman Wood's question. I've been very much thinking along the same lines. The other thing about correlation, it seems like it's easy to come up with correlations, correlation. It's very difficult to come up with a causation. And at some point, though, how do we agree on the causation. Like, some diagnoses are only by, you don't know what your ailment is until you've gotten rid of all the other things. And I'm heartened to think that the department is looking as closely at this, and precautionary approach on the screen. It seems absolutely necessary right now that you look at this and look at all of the correlations to come up with this causation. So that's all I have to say. Thank you.
Any other questions? You have more information for us, do you? While you're getting ready, Mr. Nelson passed a note to me, and so the public's aware of it. At 4 o'clock is drop dead date. Is that a good time?
Yes, that is drop dead.
Okay, because we no longer will have the facility available beyond breakdown, correct?
Yeah, Mr. Chair, we have to have all of our stuff out of here today by 7 o'clock, so meeting needs to end by 4 so that we can get broken down and packed up.
Okay.
Mr. Chair, does that mean in the next hour and a half, we're going to go through all the public forum questions and the board discussions?
I'm sorry, repeat that?
I'm just wondering if that means that we only have 90 minutes for all the public forum discussions and the board questions to go through?
I'd like to take a recess and all of us kind of, not a recess, but talk about how we want to proceed if we get close to that, which we are getting close to that time.
Okay, because there's a large amount of public here that showed up for that, I think.
What's that?
There's members of the public here that showed up for that, so I
Oh,
just
yeah.
want to be
I understand.
respectful of that. Thank
I
you.
understand. So what you would like to do is to go directly to that and skip some of the
I'm not
present-
saying that, I'm just saying we've got 90 minutes to accomplish a lot and 2 more presentations, so.
I'm soliciting input from you folks as to how you want to proceed. And I'm asking your opinion, well, how would you like to proceed on it? Just a second. Okay.
I would like to hear from the public, frankly, but.
Mr. Godfrey?
I would echo the same thing. I mean, the presentations are submitted, they're there, they're available in the public record, even if we don't get through them all. But anyone who came to speak, I would like to hear from.
Mrs. Zure?
I think the final thing where the board was going to discuss, what we were going to do, that could be put off maybe to another meeting. I don't know if it could, but I mean, that would be my suggestion, so that we could get to the public. They spent money to come here. Even the reports, we could maybe do something else with, but the public did spend money to come here.
Thank you. My feeling is we have the reports here. If individual members have questions, they can always call staff or do the research. And I would like to get to the public as well. Mr. Carpenter? Swenson's fine with that. Mr. Wood?
Missed the question.
How are we going to proceed? We're running out of time. Should we go to the public aspect of the meeting now rather than hold off and possibly lose it because we got to be out of here by 4?
Hearing from the public's pretty darn important, so I'm inclined to jump there.
Totally agree. So that's what we're going to do. We're going to throw the agenda out the window. It's not that their presentations aren't most welcome, because they are, but you heard the reasoning behind it. And we're going to go directly into the public aspect of the meeting. So we'll stand at ease for 5 minutes. We'll take your setup, or what?
We are set up.
Oh, we are set up. We don't even have to wait. So we will proceed into the public aspect. Thank you. So let me get my agenda in front of me. Hold on, folks. We have Department of Law, but we're not going to go there. We're going directly to public. If any member has a question of the Department of Law, they've presented a lot of information in the past, which is made available to you. If you have any questions whatsoever, pull one of the 2 lawyers off to the side, and they will be glad to explain it to you. With that, we're going to go into the next agenda, which is an open forum discussion. It is restricted to the subject matters that you see under the 4 bullets. We're going to take them up individually, but I'm going to combine the first 2 because both of them deal with hatcheries. So those of you who wish to address either the first or the second bullet, you're more than welcome to come to the mic, identify yourself, and proceed. It'll proceed under the same type of arrangement we do committee of the whole, which means though you're not necessarily restricted to 3 minutes, we're going to keep the comments pertinent to the subject matter. And once we start to a point where we're receiving no new information, we'll cut it off. But otherwise, we want to hear any information you have to present to this board concerning these subject matters. And I'm not sure that I can anticipate it, but it looks to me like Mr. Umphauer is ready to roll.
Thank you, Mr. Chairman. Just before lunch, I asked you if I could give the reason why we're all here today. You said I could. That won't take long. Prior to the 10th of April, 1996, the Elephant Cove put in a proposal in to the Board of Fish. I'm going to read it. That's why we're here. Proposal 421. Amend this regulation to provide the following for Prince William Sound and Southeast Alaska. Since cost recovery fish are effectively putting commercial salmon fishermen out of business by producing an excessive volume of poor quality fish, hatcheries shall be required to stay within their original mandate, which is only to maintain historic run strength on stocks that are involved with, recover their originally projection, reasonable minimum operating costs, and not to exceed that. The problem, excessive hatchery production of low-cost pinks and chums are flooding processors with huge quantities of poor-quality fish. Fish often harvested for the sake of high-volume eggs alone. Hatcheries that emphasize pink and chum production have effectively, because fish farms produce ever-increasing quantities to cover their ever higher operating costs, higher production rates also increase potential disease problems in hatchery and wild stocks. What will happen if nothing is done? Salmon fishermen are being shut out of markets by cheaper cost recovery fish. Poor quality fish reduce consumer satisfaction with Alaska salmon and results in lower overall prices for wild natural stocks as well, multiplying the economic impacts. Many fishermen will not be able to compete with hatcheries. So what they wanted to done is complete closures of nonprofit hatcheries rejected because there is a need to maintain high historic stock levels. That was the Elephant Cove Advisory Committee. And what that caused to happen is it caused the board to get with the Department of Law because the hatchery people said the board didn't have the authority. So finally, in 1997, and it's posted back there, the Department of Law gave the report. And then what the board did is they appointed a hatchery committee.
Virgil, let me ask you to go into where we go into the future, where the question is, rather than dwelling on history in the past.
Well, what resulted in this was the last thing I did as a Board of Fish member in 2002, myself and Don Coffey negotiated the joint protocol on salmon fisheries between the board and the department. That is why we're here, because of this thing right here. But there's so many different things that this, and the other thing, and the reason why Elephant Cove wrote that is real quick, 30 seconds. PISWAC decided they were going to cut into the market for the AYK wild chum salmon market on the East Coast to the John Nagel Company. So they got hold of John Nagel Company and says, we will supply you wild silver bright, they didn't call them chum, silver bright wild salmon for 50 cents a pound less. So they landed them at Whittier, sent them by train to Anchorage, processed them, put them in 50-pound wet lock boxes, turned the semi-truck down to 27 degrees, and drove them back there. They run everyone in AYK out of business, plus run the price of fish in the toilet, and that's what caused the Elephant Cove Advisory Committee to submit that proposal.
I appreciate that information. To the question of whether should independent research be conducted to ascertain what, if any, impact the hatcheries release of pink salmon may have on other species, do you believe that there should be or should not be such independent research?
We've already got the independent research done, and you just discussed it a few minutes ago, that research.
Okay. And the second question, is a precautionary approach being implemented when managing hatcheries? If so, is it working?
No, it's not being followed, and it's not working. That's why I'm wearing this damn hat. The guy sitting next to you can wear them, and Joe Spader can wear one. We're part of the guys that did that, sustainable salmon policy, of which the precautionary principle is a part of it.
Okay, I presume you were referencing Mr. Nelson. Thank you, sir.
Thank you. Have a good day.
You too. Next.
Yeah, my name is John Craig. I live in North Pole, Alaska, and I fish on the Tanana River. A global study of 51 years of peer-reviewed studies examining the interactions between hatchery and wild salmon has concluded that human efforts to improve on nature have often done more harm than good. Of the 206 studies on this subject between 1970 and 2021, only 7 reported wild salmon were actually helped by the hatchery intervention. 70% reported hatcheries had nothing but adverse effects on wild salmon. Of the studies cataloged, only 3% found hatchery programs benefited wild salmon. Hatchery fish have a higher straying rate than wild salmon. This was reported by Alaska Department of Fish and Game. Also, research shows that hatchery fish, after just 1 to 3 generations, have a much lower reproductive success in nature than wild fish. After 4 to 6 generations, average fitness is about 50%, which will inevitably lead to the depletion of the coexisting wild stocks. An argument could be made that any intermingling is harming the wild stocks. The Rigertone study that Chairman Wood referred to earlier states hatchery production in Alaska should be capped and reduced to levels that allows for the management of the wild population and no overharvest of wild populations, while also minimizing unintended competition among hatchery and wild salmon at sea. I am the first to admit I don't have all the answers, but I ask this board to use the many, many studies in their decisions on this topic. Studies show the loss of fish stocks and fish size can be remedied by lowering hatchery outputs to let the wild stocks recover. I again ask the Commission, the Fish Commission, to use the studies that are already there to make their decisions. Thank you for this opportunity to testify.
Thank you. We'll see if there's any questions. Just to the board members, while we've been sitting here, we've had a lot of RCs dropped in the blue folder, and several of them would relate to issues you brought up yourself in this, and I'm sure others will be testifying too. So I didn't have a chance to read all through them, but some of the numbers you were describing were in there.
Thank you very much.
Yeah, any questions? Thank you, sir. Next person.
Good afternoon, board members. My name is Kurt Chamberlain. I'm Assistant General Counsel for Corporate Governance and Litigation for Chillistic Corporation. As my title may indicate, I'm not a biologist. I used to be a commercial fisherman, but I got chased out of there due to the poor economic conditions in the Kuskokwim River. But in the time I had to become a trial lawyer prior to moving into my position, I ran a litigation firm in Las Vegas, Nevada, and oversaw over 50 trials and arbitrations in there. And in many of those, I cross-examined scientists, doctors, neurosurgeons, all sorts of things. And one of the things I noticed today is the standard you guys are holding the scientists to proving causation is beyond the standard any court would require. Most courts where I practiced focused on reasonable degree of scientific probability or certainty, depending on the jurisdiction. And that brings me to causation versus correlation. If you're looking for causation from a scientist, you're going to be waiting a long, long time. They can't agree the sky is blue. It comes down that they'll agree that's what light is refracted in short wavelengths. But when you break down, when you get down to proving facts, scientists work on.
Let me bring you to the two questions that are subject for this forum.
Absolutely. I'm leading into that. And so if we get to that, one of the things, the question is a precautionary approach being implemented when managing hatcheries? The answer is absolutely not. The reason I say this is if you're waiting for certainty, we're not going to get that anytime soon. You can stretch it out until the end of days and we won't have scientific certainty. The primary maxim of bioethics that you see in doctors and biologists is first do no harm. And right now we're putting our finger on nature scales in the favor of pinks and presumably at the expense of all the other creatures in the ocean. And so we do need to maintain a precautionary approach and not wait for that scientific certainty that will never happen. We do need to take, very precautionary approaches because there are something's happening in the ocean. There are multi-species collapses. And if, I heard a joke in a back area, and I don't want to repeat it and repeat the people involved in it about our fisheries collapses because it was told in confidence, and I don't want to betray that. But it really upset me when we were talking about, people managing through multi-system collapses and inactions being done. Inaction is the ally of extinction. And very quickly, Allstate in the 1990s implemented an algorithm that showed, based on research, it showed if you deny and delay a claim as long as you can make even more money. And they started doing that in the 1990s. And their daily profit from that was $36 million a day. So delaying is an absolute tactic used by a lot of people to stretch things out and delay the needed effects for conservation. One of the things we need action. We need to do it now. We've got threatened species. My river, particularly in the Kuskokwim, has been on its backside for the last 30 years. And in the last 10 years, we've seen 30% outmigration of Native Alaskans. That's loss of culture, loss of language, loss of connection to the land, loss of the safety net for our seniors. So inaction will only further exacerbate that and lead to the extinction of not only our salmon, but our cultures. Thank you very much.
Thank you. You have RC5 that your client has submitted, I presume, that elaborates on what you just testified here today. This is one of them I've not gotten a chance to read.
That's a more technical approach to that. Okay. What I'm speaking to are the specific questions 1 and 2, and more particularly 2 in that matter.
Any questions of Ms. Zeray?
Just a comment real fast. I'd like to thank you for your unique, multiple perspectives.
Thank you, Mr. Zeray. It was entirely involuntary. If I could be a fisherman on the Kuskokwim River today, I'd be that, and you would never hear from me. Thank you.
And I got a chuckle out of the reference to Allstate. Being an attorney myself, you're absolutely correct. Thank you, sir. Anyone else wish to testify in front of the board? Mr. Couch, one of my local guys.
Local to Matsu Valley. My name's Andrew Couch. I'm a member of the Matanuska Valley Fish and Game Advisory Committee, the Matanuska-Susitna Borough Fish and Wildlife Commission, and I'm speaking on my own behalf. And I feel that our precautionary approach has absolutely not been used. Like a lot of other people, a lot of other business people in the salmon industry, we've seen wild stocks go down, and we're certainly using the precautionary approach where a lot of in-river users, even people out in saltwater, certainly within Cook Inlet, their fisheries have been depleted to where some of us are not fishing or hardly fishing at all. or not allowed to harvest fish. And yet, the fish that are, the hatchery fish that are not supposed to be impacting and may be impacting, we haven't come to the conclusion that they are maybe, but those fish, we are not, in my mind, we're not managing those on a precautionary manner. If the precautionary manner is they may be affecting some other fish stocks, I think we should be doing something about it, besides closing out all the other user groups that depend on the wild fish stocks. That's about all I have to say. Thanks.
Thank you. Any questions of Mr. Couch? Thank you, Andy. Anyone else wish to present evidence?
Thank you. For the record, my name's Dr. Jim Simon. I'm a consultant for a number of intertribal organizations, but I'm speaking here on my own behalf. I'm from a multi-generational non-native family in the Otna traditional territory, and so have grown up eating some enhanced stocks from the Copper River. But I don't, with respect to the specific questions, I think that we do have some independent academic research that has raised serious concerns. I think more research is needed, but the call for more research should not be used to forego action. I don't think the precautionary approach has been implemented when managing hatcheries. Just 6 or so years ago, as the chairman of the Kuskokwim Area Regional Planning Team to develop a draft comprehensive salmon production plan on the Kuskokwim, the department's position was, quote, "For all intents and purposes, the ocean has an infinite carrying capacity," unquote. I was pleased to, at dinner with the commissioner a week ago, that the department may no longer believe that, but I think clearly we need to put more precaution, as well as consider the Alaska Department of Fish and Game's climate adaptation strategy that dictates, with increasing environmental change, we should be reviewing statutes, regulations, and policies to be more precautionary in light of those changes, which the current commissioner and I were part of helping develop that strategy. I think the question as to whether or not the Alaska Salmon Fishery Enhancement Program should be used to rehabilitate depleted king salmon runs across Alaska, I have very serious concerns about that. We are not Washington or Oregon. I think we should not give up on our wild stocks. I think we need to pay closer attention and reread the Sustainable Salmon Fisheries Management Policy and save our wild stocks before doing risky business. As the former steering committee, vice chairman of the Arctic Yukon Kuskokwim Sustainable Salmon Initiative Steering Committee, I recall when the National Research Council had an AYK salmon expert panel develop recommendations as we constructed the AYK SSI Salmon Research and Restoration Plan. One of their most significant cautions to us is that we must understand why our wild salmon populations are in decline and fluctuating before we even consider enhancement or hatchery restoration, because we could just contribute to the problem unless we understand the mechanisms at work presently. And I'll end my comments on a concern that I have about current, it's just a hypothesis. I can't prove that, the situation that I am about to explain is related to straying hatchery pink salmon. But in 2019, my family, I was not part of this fishing expedition, but my household and a household that was working for my family went to the Chitna dipnet personal use fishery and dipnetted, I think between the two households, something like 45 salmon. And when they were brought home and I started to work them, I was quite disturbed to find 20 pink salmon in the catch, and
did a little screaming and yelling about what the hell are these pinks doing in here. And I think while I don't know that those were hatchery strays, my understanding from indigenous knowledge is that is not a very common experience. I don't know what was happening in 2019 with some of the drought conditions, et cetera, your scientists have recently mentioned. But it does greatly concern me that those pink salmon that I ended up having to process instead of a nice Copper River red had gone by Miles Lake sonar and were enumerated as sockeye passing the sonar. And it leads me to the hypothesis that the reason why we continue to have not met amounts reasonably necessary for subsistence in the Gokona upriver portion of the Upper Copper River drainage continue to not be met based on the ANSs that this board has adopted could in part be due to the fact that the enumeration and management, in-season management of the Copper River sockeye run is being compromised by hatchery pink strays. So I think that's an example of where precautionary management principles need to be brought back into the picture here, as fortunately Member Umfenhauer and others who helped create the Sustainable Salmon Fisheries Management Policy and the protections of our wild stock have intended in the past. Thank you very much.
Thank you. Any questions? Thank you. Excellent testimony. And thus far, I'm really impressed with the quality of testimony coming in front of us, and I hope it continues. Next up.
For the record, my name is Shannon Earhart. I am a Navajo tribal citizen, resident of Fairbanks, Alaska. I let everybody know I work for Tamna Chiefs Conference, but I'm here on my own. And I'm not going to get near as scientific as everybody else. I want to say for your first question about should independent studies be done, I think yes, definitely, because we have a lack of confidence in what we have been hearing and seeing. And as Alaska Natives of the interior, we are being affected. I also want to say that our indigenous knowledge and the people that live out there should be included in these independent studies because they're the people that are seeing what's happening out in the lands and the oceans and rivers. As far as the precautionary approach being implemented, that's no, and nothing has been working as we've been kind of hearing because things are happening out there and we need to take a common sense approach. We cannot, I hear the science, and we cannot keep waiting and waiting because scientists don't agree. They agree to disagree. We have to use some common sense, practical knowledge there when we're going to make a decision and take a stance and let's take some action. That's what we're asking for. And I just want to thank you all. I know it's a hard job, but let's do some common sense and move forward and take some action. Thank you.
Thank you. Shannon, this is a small state. You of all people know that. But I have information that your organization is bringing in some of the experts that have been discussed here today, and hopefully we'll be coming forward with some really specific suggestions as to how to go forward. Thank you very much. And I don't have any questions. I just want to say keep it up. Any questions? Thank you. Anyone else wish to testify?
Thank you, Mr. Chairman. Dr. Joe Spader, I serve as the research coordinator for the Bering Sea Fishermen's Association. I'll try to keep this brief and focus on the RCs. The Bering Sea Fishermen's Association submitted RC number 2, which is the Greg Ruggiero paper, which has been referred to previously. RC number 3, a global synthesis of peer-reviewed research on the effects of hatchery salmonids on wild salmonids. That's the paper it's been referred to previously. 104 papers. 83% found either an adverse or a minimally adverse impact. The take-home message of this is, we've been talking quite a bit about causality and correlation. It's fairly rare in in the life sciences that you have direct causation because we don't have multiple replicates. The way science operates without mechanistic explanations or direct causality is the weight of evidence. In terms of a weight of evidence, this review provides an arguably compelling weight of evidence in terms of drivers' negative adverse impacts of hatcheries. RC number 4 is a kind of a straightforward way of trying to array the adverse impacts of hatchery salmon across the life cycle. And many of the papers in the Macmillan et al. Are actually focused on Pacific Northwest and other regions, but don't think that they don't apply to Alaska. The genetics associated with the deleterious effects within 1 or 2 generations applies everywhere. So in response to the question that's before us, that's particularly related to the last question, or second to last question, should the Alaska Salmon Fishery Enhancement Program be used to rehabilitate depleted king salmon across Alaska? We would argue that no, it should not be applied. One of the most compelling arguments is there's a very different of applying hatcheries in pristine environments versus existing hatchery operations. Virtually all of Prince William Sound is depends almost entirely on hatchery stocks. The Hatchery Scientific Review Group created by U.S. Congress addressed this question in particular. They said hatcheries are used to boost harvest throughout most of the native range of Pacific salmon, including regions that are relatively pristine and that support sustainable wild salmon populations. From a science and precautionary perspective, the Hatchery Scientific Review Group recommends 0 hatchery releases in regions that are relatively pristine, and that the best management practices in pristine regions should ensure that relatively few hatchery origin salmon spawn in the wild. Otherwise, our comments are found in RC number 6, I believe it is. And lastly, I will just leave you with the fact that, again, the sustainable salmon policy requires a precautionary approach. And we've talked much discussion about uncertainty because lack of causality, direct linkages. But the sustainable salmon policy is quite clear that where the impact of a resource is uncertain, but likely presents a measurable risk, priority should be given to conserving the productive capacity. And lastly, it says the appropriate placement of the burden of proof on the threats or the activities that pose risk. I would submit that the burden of proof is backwards here. You're being, a variety of scientists and others are being asked to provide the burden of proof that wild salmon are being threatened by hatcheries, when in fact, arguably, the weight of evidence, which is our only meaningful standard moving forward, suggests that there's powerful evidence of the impacts, and that the burden of proof should be turned the other way. Thank you.
Any questions? Mr. Godfrey?
So the burden of proof should be flipped around, you're saying. And so, in essence, you're saying the burden of proof should be hatchery fish. It should be proven that hatchery fish are not harming wild stocks, is what you're saying?
That's correct, through the chair. But I'm not saying this. I'm reading from the Alaska Sustainable Salmon Policy. That's the policy that should be guiding these kinds of questions. And also, that's the policy that provides the Board of Fish with part of your intersection with this very issue. That would be the precautionary approach that Mr. Templin also referred to, that the department acknowledges. But I think there are, I think, yes, the burden of proof, especially if there's any consideration to hatcheries in pristine regions such as Western Alaska, the burden of proof should adhere to the Sustainable Salmon Policy.
So, and thank you for the answer. So, do you believe that's actually an attainable approach? Because I'll preface it by saying that absence of evidence is not evidence of absence, and proving a negative is virtually impossible. So, in your mind, that would actually be attainable conclusively?
So what can inform action in an environment such as Western Alaska, where there aren't currently large-scale hatcheries? We commonly in science, we would use the weight of evidence. What is the weight of evidence that hatcheries that have been implemented in other pristine environments are not likely to cause harm? And again, many of these issues, particularly related to the deleterious effects of multiple generations of hatchery fish, which has been discussed earlier, that occurs even as early as within one generation. They have found, this was with steelhead, about 755 genes were altered within one generation. The point being, the genetic issues are independent of the location. That's just simply the effect of taking a wild brood stock and placing them, basically skipping mortality in the early life stage. So there's nothing magic about hatcheries in Alaska that they somehow can avoid the deleterious genetic effects.
Thank you.
Anything further? Any other questions? Just have a real quick couple of them. I think I know the answer to this, but what do you have your doctorate in?
I'm sorry, I didn't.
Didn't you identify yourself as Dr. Spader?
Correct,
Okay,
yes.
what is your doctorate in?
PhD in ecology from University of California, Davis.
Thank you, and you've practiced in that field for some time, have you? What I need you to do is just for putting on the record, and I'll preface my remarks by saying all of us come from a different world, and I come from a legal background, and you can determine in court, and it's not all that uncommon, consistencies. In other words, is the stab wound consistent with a serrated knife, et cetera, et cetera. And if you're a defense attorney and you get too many of those consistencies, all of a sudden you find your client sitting in jail under beyond a reasonable doubt, which is the highest standard. So the correlation arguments, though I understand them, are not going to carry the weight of the day for me. But I need you to define for the record what the weight of evidence is as far as a test? How do you know that you are at the level that warrants action in a certain way? Does it have to be a 51-49% split, a 60-40? Could you please elaborate on that?
Yes. The weight of evidence, it's essentially you could say a tipping point within the academic community. There's many topics from quantum physics to salmon ecology where there's difficult questions, and once there is a weight of evidence, there is a tip within the community and a broad consensus.
How would I decide that we've reached that tipping point?
Well, one example I think is arguably RC number 3, the McMillan et al. Paper, which also includes Dr. Rugroni, which is an absolutely comprehensive survey of the literature. An example also is carrying capacity in the Bering Sea, for instance.
So it's a level below what we've been referencing as causation, basically.
Well, we don't have replicate Gulfs of Alaska or Bering Sea. And that's why if we don't have replicate systems, we have to rely on correlation and the weight of evidence rather than direct causation.
Okay. Appreciate it.
I hope that that helps, but I don't think there's a magic number that can guide these questions. But it's the preponderance of evidence, to use a legal term.
I understand it fully. Thank you. Any questions? Thank you, sir.
Thank you.
Anyone else wish to testify? Going once. Yeah, I thought I'd call somebody's bluff.
Yeah, hi, my name's Nancy Hillstrand, and I'm a seafood processor on the end of the Homer Spit. I've been in business 32 years, and I also spent 21 years as a fish culturist with the Department of Fish and Game, and did a lot of habitat work back in the 70s, 80s, and 90s. So I've learned my lesson now, and after being a fish culturist, I realized that, yes, we do need an independent panel to do research, kind of like the 3 wise men. Like right now you have the one wise man, but I think we need a couple more wise men to help for you to consult with, so you can see the wisdom there is in the world on this issue that's not quite so close to the politics of Alaska. An independent research would be Socrates' art of argument, and we need to be able to argue and not be mad at each other. Just the main point here is we need to protect the wild fish, and we're losing them. And so anything we can do, there's multiple things harming the wild fish right now, but they don't deserve for us to keep adding more things on top of them. As for the precautionary approach, 39-222, the sustainable salmon policy, that was put together through a 4-year period, and we could create the same type of thing with a research panel using maybe that same format that would give us this counsel that we need to do the right thing. But I do believe we need to do things ASAP right now, and I think mine's RC3, where I've listed just some ideas that we could start working on, some of these things that we should start doing right away, such as defining some of and adding that to the sustainable salmon policy, because not everything is defined in there, so we need some more definitions so everyone is on the same page and standardized. I'm concerned about the AHRP because in 2015 they stopped taking any chum salmon out of an index stream called West Crawfish, and it's one of the largest streams in the outer northern southeast district. And since that time, they started doing a remote release, and that's a real important word to remember, is remote release site, because that is something that comes up in what's called a PAR, which is a permit alteration request. And when that comes up, they just, they can put 20 million fish here, 20 million fish there, and they go into these pristine areas. And what happened at crawfish is it started to stray in massive amounts, up to 99 to 100% of the fish in there were hatchery fish. And this is an index stream that's used for enhancement goals. And so what is happening with our enhancement goals when they're getting bloated by a hatchery fish? So we have to start looking at our escapement goals and monitoring them, which there's no money to put into this. So that's something that maybe the board can help get more monitoring of otoliths out of these enhancement goals, so we know that they're not being overrun by hatchery fish, which makes it look like wild fish are doing okay in those streams. And the other area, of course, is the mixed stock fishery, which is a place where the wild fish are being scooped up in a hatchery harvest. And so we don't know unless we do massive monitoring in those areas also. So Southeast is losing their chum salmon, the wild chum salmon. They have hatchery salmon, but that wasn't shown on the presentation. But I think it's really important that you look into what are the wild salmon doing in Southeast. As in British Columbia also, they're going down. So what can the Chinook, trying to use hatcheries for the Chinook, I think would be the last thing we would want to do for our beautiful wild Chinook salmon. And the first thing we want to do is find out all the factors that's causing the problems here. And we're doing that right now with Ed Farley's task force, which I'm really happy we're doing that, but we need to start acting on whatever the low-hanging fruit is so that we can start to help these fish and find out where the blockages are. Habitat Division has a lot of information on, for instance, on the Susitna, there's 150 blocked streams blocking salmon spawning areas, and we need to start, rehabilitating those areas as soon as we possibly can. And so the other thing is the ADF&G, how to better communicate. The first thing ADF&G could do is to be more honest with you, because when I listen to some of the things that are being told to you, it's not that they're necessarily lying to you, but they're not bringing all of the information that you need, or they're going around the corner of answering your questions. So that, of course, comes back to having a nice panel that can offset some of these problems so that you get the best possible information that you can get for making regulatory decisions. Since 16440B, you've heard of that for the authority of the Board of Fish. That has been taken away from you by the Department of Fish and Game. And in 2021, I put in I guess it's PC7, which shows that 5AAC 40.480, it's called a notice of permit alteration. It's a notice. It's not a means of the department being able to modify. It's a notice that lets the regional planning teams get a hold of what the industry wants. They negotiate what they think is the best thing to do. They make a recommendation to the commissioner, and then the commissioner is supposed to consider and make any other recommendations, and then it was supposed to come to you by statute. And so you might want to look at that regulation, because it was just changed, and E was put in there that actually took all of the information, or took all of your power away from you, so that you can no longer, you still can because you have the statutory authority, but by regulation, they've kind of bypassed that, and I think that's a wrong thing for the department to do. So please look at that. Anyway, I just hope we can get any baseline information on crab and shrimp areas where some of these hatcheries might be going into, because we're releasing these many fish into these areas. For instance, Prince William Sound from 1960 to 1976 had a harvest of about 4 million fish, and that weighs about 12 million pounds of predator biomass, eating approximately 500,000 pounds of food a day. So that comes out to 15 million pounds in a month when they're milling around ready to spawn. Well, now, since 2010 to 2019, this is about 40 million, so it's 10 times the amount. That comes out to 120 million pounds of predator biomass in the nearshore waters, and that means 150 million pounds of food in the last 30 days. So we have a great big, huge sea otter out there, and if you want to consider it being a predator, it is a huge predator. It's a wall of predation, and it's in our nearshore waters, and it comes every single year, and we just need to, the precautionary approach, we need it so badly to protect our fish. So I'm all for wild fish. I'm not allocative, but I really, I beg of you to take care of our wild fish before they completely go down. We're one of the last places on earth. I mean, my God. So anyway, thank you very kindly.
Thank you, Nancy. Before you go, you've got 20-some-odd years of experience in the department. How many of those was spent in Fred and dealing in hatcheries?
I always stayed in Fred, and most all of it was in hatcheries, unless, I mean, I moved around between 3 hatcheries and all these different habitat issues like Chinook and Sitka-Sulak up in the Arctic and down south end of Kodiak.
Okay.
And so I got a really good experience with many different places and multiple out migrations and incoming adults, so it was pretty.
Okay. Any questions of Ms. Hillstrand? Mrs. Zurui?
I'll try to make this quick. I really appreciate your comments on that independent research idea, and I'd like to talk to you more about, how you'd maybe set that up. There was I believe you were talking about something that would be equivalent to something I was trying to get at earlier with some questions, and that is terminal harvest areas, the degree of segregation of the wild and the hatchery fish, and is there studies or anything that I could have you tell me about, or could you speak to, it seemed like you were talking about, there being a good degree of mixing of these two, the wild and.
Yeah, it's in the Mark Tag Lab has a lot of this information. It's kind of hard to read, but once you understand how to input the information you want, then you can bring up what the stray rates are all across the state. And SHAs are where the cost recovery can take place. So they're not the terminal, they're not right up against the river. And I wanted to say, I'm glad you brought that up, because SHAs are where a lot of wild fish can be caught. And for instance, like near the Tutka hatchery, it's anywhere from 5 to 10%. So if you have 1 million fish, it's still quite a few wild fish that are being caught, and they're not counted as wild fish. They're called hatchery fish, so they're not being accommodated for that, and it's not accommodated in the enhancement reports. So, but you can find that in the Mark Tag Lab information, and there's a lot of straying issues and papers that were done prior to the AHRP. And the AHRP, also, they never did really have any baseline on pinks. I think it started in 1990 when they actually started getting any baseline data on what a wild, what they considered wild pink. So that's 20 years after hatcheries had started, or 15 years, so there had been a lot of input already on there. So we don't really have a lot of good information on baseline for that, so.
Thank you. Yeah, and that was, those areas were, I just wasn't using the right language, before, the terminal harvest areas versus, that's what you're talking about was exactly what I was trying to find out.
Well, and it's in your purview also under 1610 440B that allows you to state where this harvest can take place. And, from there, then you can try to, like I say, the remote release sites, it's really important that you start looking into those, because they're they plop them down into, pristine areas, and it's getting blanketed with these areas. So they're not just the 8 hatcheries, it's another 100 remote release sites, so it's pretty dangerous. And 20 million apiece, it's quite a bit.
Any other questions? Thank you, Nancy.
Thank you.
Anybody else wish to testify? Don't wait too long, come on up. Pardon me?
I have a question for you with respect to
For me?
Yeah, for you.
Okay.
So just for the clarification, are you asking for comments or testimony only on the first 2 questions, or have you opened up to all 4 yet?
That's where I started off, but they started wandering
Okay,
to question 3 and 4.
I didn't want anybody to sit out there waiting just in case they had something specific
Yeah.
that they wanted to speak to on 3 or 4. Come on up.
Yeah,
Thanks.
thank you for pointing that out. I just let it get lax and come in. Yeah. Duncan, you have the floor.
Thank you, Mr. Chairman. I'll try to be brief in the interest of time. I work with and have represented the rural communities of Uzinki and Old Harbor for over 30 years. The fishermen in those communities have commercial fished for over 130 years. The fishery in Kodiak developed about a decade before the fishery in Bristol Bay. Having said that, we're very supportive of the hatchery program in the rural communities on Kodiak Island. We believe the hatchery program has enhanced our fisheries. We believe it's evened out the various fishery cycles that we've seen over the years. We believe that our wild stocks are healthy and have been maintained. We see year in, year out benefits of the hatchery program on Kodiak Island. It seems as though we've developed a single narrative here today, and I wasn't planning to testify, but I felt compelled to get up and say there's a significant other perspective, even among Native Alaskans, particularly those that have actually been engaged with the hatchery program. Having said that, Mr. Chairman, just very quickly to your 2 questions. The first question relative to independent research implies that somehow the current research the Department of Fish and Game is doing is inadequate, inaccurate, or mistrusted. That's not my point of view. My point of view is the Department of Fish and Game does an excellent job in research. They're working very hard to find answers to these questions. Of course, independent science is always helpful, but I don't believe that there's a bias with the Department of Fish and Game, and I believe that question implies a bias, Mr. Chairman. Moreover, the question only addresses pinks, and I think a fuller discussion should address both pinks and chums. That perhaps is just an oversight in the question itself, but we need to consider the impacts of both species. With regard to the second question, Mr. Chairman, the precautionary approach that we have is self-evident in the relatively stable releases of hatchery fish over the last 30 years. We have had a significant precautionary approach because you had a very rapid curve from about 1982, 84 to about 1992, and then it's been relatively even. That's a precautionary approach. Is it working? Lots of questions there. Is it working for whom? Is it working for what species? Is it working for what region? All those things. But in terms of, is it working that we have maintained both wild stocks and hatchery stocks in the areas where the hatcheries operate, Southeast Alaska, Prince William Sound, Kodiak, Cook Inlet? The answer is, for the most part, yes. Do you have depletion issues in the Mat-Su Valley? Yes. Do we have information that pinks and Prince William Sounds are related or causally related to those issues? No. In fact, this is what was particularly interesting today, is the spatial separation that we saw in the first presentation relative to chum salmon in the Gulf of Alaska. In the Western Gulf, there are very few hatchery-released chum salmon from Alaska relative to those released from Asia, particularly Japan and Russia. In fact, it's less than 10%. It's more like 6% of hatchery releases from Alaska that impact that. We could eliminate all the chum hatchery releases entirely, crash our program, and not make a dime's worth of difference in Western Alaska relative to the mixing of chum salmon with, in the Western Gulf of Alaska, where they're surveying the winter migrations of chums. We wouldn't make a difference relative to the impacts on the chum in Western Alaska. I understand the concerns from those in Western Alaska. Huge issues, huge problems. We don't have the answers, but from what we do know, the answer is unlikely to be chum releases in Southeast Alaska. I really think that we need to focus on that spatial distribution. And by the way, where were the pinks? Oddly, those pinks were not interacting with the chums, coho, sockeye, that they surveyed on those surveys. Those pinks were somewhere else. So where's the causal relation there? We're not talking correlation. We're not talking causation. We're talking absence. I've gotten wound up, Mr. Chairman. Very important to parse these complex issues out by area, by species, by time, by multiple variables. Before we move forward to talk about reducing the amount of eggs that hatcheries are able to incubate and release, we need to really, really dig in to the details and great philosophical discussion. Do hatcheries impact wild stocks? Move from that to the specifics by region, by time, by fishery, and perhaps we can find some solutions. Thank you for your time, Mr. Chairman.
Thank you, Duncan. Any questions, Mr. Fields? Thank you, sir. Anyone else? While you're coming up, it is 3:33. Just.
All right, I'll go fast. So my name is Patrick McCormick. I'm an Area E drift gill netter. I'm also a sport fishing guide. I also work for the Department of Fish and Game, and I'm a science teacher here in Anchorage. I deal with hatchery fish every day of the summer. They're the lifeblood of our fishery, especially in years when the Copper River is low. And my big concern with hatchery fish, there are all these environmental impacts that we are starting to see and understand, and we need to continue studying those. For me, one of the things, though, that I want to address and have studies for people smarter than me is the economic impact of the hatchery fish beyond there are this many more fish. The fish that I catch that are hatchery fish are tiny. I can tell a pink salmon from Wally Nornberg because it's 2.5 to 3 pounds. I have to buy new gear to catch them. The average size of pink salmon in places without hatchery pink salmon are like 4.5 pounds. And for me as a gill netter, when I have to take every single one of those fish out of my net, it makes it harder for me to make money. So as weird as it sounds, and as much as I assume most of the fleet disagrees with me, it's hard for me to make money with those hatchery fish there. In fact, in 2020, we were limited in our ability to fish for wild fish because the hatcheries could not get brood stock. 2020, I drove part of the Southwest District, and it was the most salmon I have ever seen in my life. And I worked in Bristol Bay counting fish. Like, I have seen a lot of salmon. It was an insane biomass that was left unharvested, and if you read the season summary reports, they even note it's left unharvested. And that year was a disaster year. There was a disaster declaration, and probably in like 5 or 6 years, we might get a little bit of money for that. But there are fish that were wild, that were unharvested because of our hatchery policies. I think it's really important that we study the impacts of the small fish and start thinking about the biomass rather than the number of fish. I also think it's really important that we reopen our strategic plans and use the prevailing research to make the strategic plans for hatcheries better, because there is a way that we can use research to make our hatcheries better. And so with what the board can do, which is allocate, right? There's not too much you guys can all do about that, but I think the more voices we have amongst those of us who care and depend on fish to make sure we're always reviewing our best practices like we're doing now is really important. I think the Prince William Sound Strategic Plan hasn't been touched since like 1996. It's off the top of my head, it's something like that. That's like a long time ago. I was 8 years old when that happened, and those decisions now are affecting my livelihood now, and it's hard for me to make money, even with the amount of hatchery fish out there. I would prefer to shift away from pinks for higher value fish like kings and silvers. Obviously, as a gill netter, I would rather catch 1 king salmon than 1,000 pounds of pink salmon. Seems pretty basic. That being said, to answer the question, should we use hatcheries to help depleted stocks? The only place I'm aware of in the world where that has helped was Redfish Lake in Idaho, where they were down to 4 sockeye. We should be using hatcheries to enhance and provide harvest opportunities. So I would be supportive of something like shifting some of the resources from producing pink salmon, which are very successful on their own without hatcheries and very easily sustainably harvested, to something like Chinook salmon in in terminal harvest areas, where we can then, reap the benefits as a whole state, not just me as a gill netter or somebody as a seiner. Everybody wants to catch kings. So policies like that, I think, would be really good. The last thing I'll say is that it's important to think about market conditions. And as we know, we had a collapse of market conditions this year. This year I caught twice as many fish as last year and made half the money. And when we're planning for hatchery releases for something that might be affecting wild fish, we need to make sure we aren't causing an oversupply of the market. Because for me, I want to be efficient. I'm pretty lazy. I want to catch fewer fish for more money. And I think if we do the right kind of research, that's the way we can do it. Thanks.
Thank you. Any questions, Mr. Godfrey?
Yeah, thank you for your comments. Did you say, in the first couple minutes of your comments, did you say that you had to buy smaller gear to catch the smaller pinks?
Yeah, I bought a 4.5-inch net, which cost about $8,000, and I also had to buy smaller gear to catch hatchery sockeye.
So if you like that category of fish less, why would you just have your gear accommodate and target it?
Because that's what there is. Like when we have problems with the Copper River, I'm forced to fish along with the rest of the fleet in Prince William Sound, in Main Bay specifically. The Main Bay fish, when I was gill netting in 2006, averaged 5.5 pounds. They average 3.5 pounds now. So clearly there's something going on there. I have my theories, but yeah, I specifically have bought much smaller gear. And
so if you didn't get the smaller gear, you would be catching nothing?
I would be catching probably, I would still catch some fish, and they would be mostly big, beautiful wild fish that I prefer. I'm not saying I'm opposed to hatchery fish. I'm saying that specifically pink salmon are very small and hard to sort through if I'm, say, targeting coho or something. They still do get tangled, you still catch them, but specifically I have a net to try to target pink salmon sometimes.
So, and I'm not trying to get you. I'm trying to understand, because you're the second commenter that's talked about kind of the economics when it comes to the value of the fish and these types of things. And so I'm a big free market person, and I think if it's as bad as some people make it sound, then you're going to get different price for hatchery fish versus wild because the quality is there. Eventually the market's going to demand that. If it's not demanding it yet, it's not that bad, from my opinion, not being in the game anymore. But if your only alternative is to get smaller gear, mesh, to catch the targeted species, but you're complaining about the species and the alternative is nothing, it's not a zero-sum game. If the smaller hatchery species were not there, more wild stock would be there. That's not what you're arguing, and that would be counterintuitive anyways, right?
Yeah, I guess what I'm saying is we should be making sure our priorities are for fish that are, marketable and that are helpful for the most number of people. I'm not going to sit here and say we should end hatchery releases tomorrow. I think that would be a terrible idea, and it would hurt a lot of people. What I'm saying is we need to deeply investigate what's going on and tailor our policies, just like our genetics policy, our strategic plan of where we're going to release fish, to make sure we're using best practices. Because to watch To have
Let me
to,
ask you to bring you to a conclusion.
Yeah.
We have other people that may want to testify as well. So, if
Yeah.
you could wrap up your
oh
comments.
yeah, so if that kind of answers your question, it's, you know, what I was trying to do. I was trying to make money as a fisherman, and I think specifically hatchery pink salmon and Prince William Sound have made it harder for me to make
And
money.
you're not at any point, you weren't implying, and I'm not saying you were, I'm just trying to be clear.
Yeah.
You weren't implying that the smaller hatchery fish are supplanting the feedstock available that's then competing out the wild stock in exchange, right? You're not
Well,
saying that at all.
I don't know the answer to that. When I look at the research available, I would say that is a pretty strong correlation, and specific to hatchery pink salmon. So that's something that I think it's really important that we take a precautionary approach when we deal with, are we going to stock more pink salmon? I also think we need to be managing on biomass, because what matters to me is my poundage. It's not the number of fish I catch, right?
Thank you.
So,
Thank you.
there you go.
Thank you. Any other questions? Seeing none, thank you, sir.
Yeah, thank you.
Anyone else wish to testify here tonight? And we'll make sure that following Ms. Carlson Van Dart's observation, you can go ahead and address items 3 and 4 as well. And if you've already have done that, don't come back up for the same subject matter. So anyone else wish to testify? You wish to testify?
Thank you for the opportunity. My name is Susan Doherty. I'm the General Manager for Southern Southeast Regional Aquaculture Association that functions out of Ketchikan. I would just like to encourage this board, if you've heard some testimony from the audience today and it resonates with you, I would implore you to reach out to the associations and ask if some of the information you have heard is in fact factual. Just one example, SARA, we go by the acronym SARA, operates 6 hatcheries and 6 remote release sites, not hundreds. That's one of a myriad of statements that have been made that may not be based in fact. We also released 220 million chum, which every single one is otolith marked. We, at the association's cost, monitor all of the fisheries in southern Southeast Alaska for the presence or absence in every fishery for every stat week that we are able to sample. We monitor, we provide that data to Fish and Game of the release location and the percentage of hatchery and wild fish in the common property fishery. To my knowledge, and I've worked for the association for more than 35 years, there has been less than a handful of times where we have not had an otolith marked fish recovered in our terminal harvest areas or Shaw areas. If we get one that was in an area, let's say Neats Bay, that isn't a Neats Bay mark, it might be an Anita Bay mark that was a release at one of our releases. So it is a hatchery fish. So there's a lot of information that you've been given that may or may not be factual, and the associations have a hard time planning for this when we don't have an agenda and know what the questions might be, what you might be interested in, and what the public might be interested in. So that would be another helpful to get accurate information at these meetings if we know in advance what you are interested in. And we plan to send you some information of what we've heard today. And please ask us if you have questions, and we'll give you the data we have that either supports or doesn't what you've heard tonight. Thank you.
Any questions? Thank you for the suggestion. What's that? I do have one. Okay, Mr. Godfrey.
While you're up there, is there any other mischaracterizations that you want to address that you've heard over the last 3 days? If there is, I just want to give you the moment, I just want to hear it, but if not, don't worry about it.
I would rather have more time to think about them. There was quite a few things that have, over the 3 days, that could be addressed.
Thank you.
Any other questions? Thank you, ma'am. Anybody else wish to testify? Gentlemen, why don't you come up here? We're not going to argue over who comes first or last.
Well, I'm always last.
Paul, go ahead.
Thank you for letting me speak. I'm going to be really close, really quick here. So when I look at hatcheries in my vernacular, being part of the hatchery associations, it's mitigation. We look at mitigation as the reason, at least as the way I see it. For instance, many of the fisheries in the Southeast, and I'm not going to speak for them, or off the coast of Oregon or Washington, Idaho, they're mitigation facilities. That's what makes this thing continue to go. So I would caution to think that you have to wait until the ultimate answer, until it's all gone, to try to start again. That is the recipe for mistakes. It did not work. But when you're talking about small-scale operations, when you talk about habitat enhancements, when you talk about surgical applications, when you talk about communities getting together and improving their situation, that's a win-win situation. You're doing something. It's when you're on a full-scale production and you have to produce a certain amount to pay for your efforts. That is a problem. Fred Division, once it changed, then the state and the community was no longer part of funding those operations, created this void, created this situation. So I just want the board to think about it. There's a lot of things out there. As an Alaska Native, we're looking at mitigation things to do, things to make it better, and it may work, but we need the support. We need your support.
Okay. I don't know if you put your name on the record. It's Paul Shadura, correct?
My name is Paul A. Shadura II, yes.
Okay. Any questions? Thank you, sir. And the gentleman who was reluctant to come up a moment ago.
Oh, I wouldn't say I was reluctant to speak up. I have no fear on that. I was just trying to be polite. So I want to speak in my personal capacity on question number 3. The release of Chinook is a rehabilitative measure on that one, and Chilista hasn't developed a position on that, so I want to speak in my personal capacity. With respect to that, this is very dear to me. You know, I was in the Kuskokwim River. We used to, with a rod and reel, catch 20 a day, and it was amazing. But as Commissioner Vincent Lang testified before the Senate Resources Committee, when you introduce a hatchery species into a river, what you end up with increased harvest of the salmon, of the resource, and what that'll invariably lead to is increased harvest on the wild resources. The wild resources on the Yukon-Kuskokwim River are in dire straits right now, and the last thing we need is increased fishing on those measures. And I also wanted to talk to the cost-benefit ratio on there. There's a study, annual cost of wild salmon restorations in the Columbia River Basin. It's stated in 2004, the cost per salmon to get a Chinook into those rivers was $400. I heard a recent study, and I can't find it right now because my laptop died, is $600 per fish. If the state wants to spend a lot of money, rural infrastructure would be great. There are 3 villages in the Kuskokwim that are having electrical power crises. Crooked Creek, Tuluksak, and my hometown of Aniak. When I was in Tokyo earlier this year talking salmon, Dr. Hayo Aiken from UAF described the Bering Sea as a research desert compared to the other areas. If we want to spend money, I strongly agree that an increase in funding there would be very much appreciated because as Commissioner Vincent Lang stated once before, the problems with our salmon don't arise within the fresh waters of the Kuskokwim and the Yukon. So looking into the Bering Sea would be very much appreciated. So I think that money can be spent better elsewhere with better results and less harmful results to our native salmon. Thank you.
Thank you very much. Any questions? Thank you, sir.
Thank you.
Anyone else wish to testify on items 3 and 4? Come on up, Virgil.
Okay, one thing I didn't say earlier, and that is that when we wrote that Protocol, Dan Coffey and I, along with the Director of Commercial Fisheries, and what the board had decided back in the late 90s is that we needed to not have what happened today happen anymore. And what happened today, it was announced when they gave the lady gave the briefing on the hatcheries that they increased production by 40,000, I think it was, no, 40 million chums someplace. That was done without no public notice. It was to have a statewide perspective, reread the sustainable salmon policy and reread the protocol between the board and the department. We wanted a statewide perspective so the board knew what was going on, and so that instead of just the regional council, or whatever you want to call the Regional Aquaculture Association, cannot make all these decisions with no one else knowing it's going to happen or even being considered, because there's no statewide public notice. What should happen is before any of them can do any increases in production, it should be published in the book. It should be published in the proposal book that gets published every year. It should be published there so that it can be presented to the board and the public can comment on it. That's what needs to happen. That's the only way you're going to solve this problem, where you won't have people like me just mad as hell because they did this again. Because the salmon truly are, the ocean pasture is truly overgrazed. I'm absolutely confident of it. And then when it goes to the precautionary principle and stuff like that, I wasn't trained to be a scientist. I was trained to be an infantry officer. No, I'm serious.
I know you're serious.
And you have to make a decision. You need to make it.
I'm going to stop you for a moment and ask if anyone else wishes to testify. If there's no one, I'll allow you to continue. But if there are people, I'm going to give them a chance. You've been up here once before. Does anybody else wish to testify? There are other people. So please take a seat, and we'll take the person who hasn't had the opportunity. Duncan, you have testified also. Let me have this lady in the back that has not, and then I'll get to you.
Address question 4 in one sentence. We need information to contextualize the value of Alaska's fisheries. Less than 10% of the salmon produced in the world are Alaska's fish. Okay. Less than 10%. When we talk about the economics, we talk about market impacts, we talk about all these concerns, we have to put it in context. So with regard to how can the department communicate accurate information relative to the hatchery program, you need additional information of how all cumulative Alaska fish.
Thank you.
Thank you.
Ma'am, if you wish to come up.
Hi there, Tina Fairbanks, Kodiak Regional Aquaculture Association. It's hard to come forward as a representative of an aquaculture association, even at a hatchery committee meeting. And so, but I think that days like today illustrate the importance of following through with the protocol in terms of talking about questions 3 and 4. And while it's easy for a lot of individuals to come forward and sort of strafe the programs or look for easy outs on some of the complex questions and issues that address Alaska salmon. I think it's still important that we have this process, and I don't want to discount the passion that people come forward with when they come into this arena. But I would just ask that, as with what we see in the caution that the department utilizes when it makes its statements and its presentations, that this board exercise the same level of caution before considering any action that affects programs that are longstanding, that have a history that's backed by science, and that is rich in data that rarely gets an opportunity to be aired for itself. And so I would ask that this board not discount the work of the department. Much of the information that they have that's relevant to some of today's questions was not given an airing today. And I'd like to acknowledge that as well and thank the department for the work that they have done to provide an unbiased approach to the questions that have been posed about hatcheries in Alaska. So I think that the hatchery protocol and the implementation of it is important to this board process, to the public process, regardless of whether or not the bulk of the opportunity for comment typically comes at hatcheries from a negative perspective. It is important to the overall discourse because it gives us an opportunity to air those concerns, for them to go onto the record, and for the quieter mechanisms of what happens at the level of the aquaculture associations and within the Department of Fish and Game have an opportunity to answer that. And so I would just thank you and hope that we can continue this process.
Thank you. Any questions? Ms. Carlson-Van
No questions,
Oh, no
Mr. Chairman,
question?
but if you're, I do have some comments when you concluded
Okay.
the
Yeah.
public portion.
Okay. Thank you. Anyone else that wanting to testify that has not? Seeing and hearing no one, we conclude the open forum discussion. Now, what is it you wanted to do?
Well, Mr. Chair, I kind of just wanted to take a quick second, and I know we're under the gun, to sort of synthesize some of the things that I heard today, if I may. So with respect to question number one, my takeaway from the discussion is that there is plenty of research that exists out there. I definitely heard that there was collective agreement that it warrants more, but I also heard a message of urgency in the testimony that we heard here today, and that we, and by we, I mean Alaskans, the department, the board, shouldn't be deterred from taking management or conservation actions, by waiting for the issues to be, air quotes, settled or sort of studied into irrelevance. So I just wanted the public to know that was some of the takeaways that I heard from what you shared today. With respect to the second one, is the precautionary approach being implemented? Yes and no. I heard a mixed bag, but Andy, who has left, kind of said something that made me think about it in a different way. I was really looking at the precautionary approach through a lens of hatchery production. But some of his statements about, there's cutbacks for precautionary conservation across other user groups. And so maybe we're looking at it a little too myopically, and that the precautionary approach should, may or may not be being applied equitably. I think in his comments, he didn't feel like it was being applied equitably, and I thought that was kind of an interesting take on it. With respect to 3, what I heard was overall a really healthy skepticism and yet an unwillingness to sort of sanction king salmon enhancement as a tool or a solution to the production issues that we're seeing with king salmon. And then lastly, with 4, and certainly with exception, I also heard varying degrees of skepticism about how the department and what the department is communicating, and varying levels of confidence in that information as well. And I appreciate the operators or the associations being here very much, because I think the perspectives that the last 2 testifiers brought were useful to me, but perhaps it's also incumbent on the associations to consider how best they, in conjunction, maybe or not, maybe independently, are putting out information. And I'm sure you do that regionally, but maybe there's a better way to do that statewide so that you are getting accurate information out directly to the public. And maybe, Mr. Chair, what that looks like is perhaps a supplemental meeting to this one at some point, where we can conclude some of the agenda that we didn't get to today, and also have an opportunity to hear from each one of the regional associations themselves, instead of it being filtered through the department. I mean, not like, but just.
That's an idea we could discuss.
But yeah, I mean, I just kind of wanted to throw some of those things out. And thank you, everybody, for your patience today and for your participation. Appreciate it.
Anything further come before the board? 4 o'clock, we're under the circumstances, we no longer have a facility. I would entertain a motion to adjourn.
Move to adjourn. Second, Saray.
Any objection? We stand adjourned. Thank you, everyone.
Wrapping it up.
Huh?
Thank you for giving us the time to back up and get out of here.
Yeah. You know, there's only, I think only one thing I hate worse than liars.